make it more comfortable. Okay, cool. Let's get off the stage.
Good afternoon, everybody. Thank you very much for coming today. I think we'll make a start. Can everyone hear me okay at the back? Yeah. So I think we'll make a start. My name's Jonathan Schott, and I'm based here at UCL at the Dementia Research Centre. And it's a great pleasure on behalf of myself and co-organisers, my colleagues Nick Fox and Catherine Mummery, to invite you to welcome you to this symposium about disease modifying therapies for dementia. Everyone in the field has been waiting for new therapies for some 20 years, and the prospect of some new therapies that we'll be able to deliver to our patients has never ever been closer, with the results of positive phase 3 trials and drugs that are now licensed in the US. So this is clearly a hugely exciting time for patients, for carers, for clinicians and researchers. The major advances and the opportunities this have are obvious to all, but this also comes with some significant issues around the practicalities of delivery of these treatments, how we can do so equitably, how we can do so safely. And there's ongoing debate within the community, of course, about the results of trials and the risk-benefit ratios. So the idea behind the symposium today is there's been a lot of debate, particularly around academics, around some of the clinical trial results and their implications. But what we wanted to do today, and the way that we've planned this symposium, is to have 3 sessions. So the first session today is to review the evidence and the implications for research. The second is to hear from someone who's been in one of these clinical trials, something that's been distinctly absent from a lot of the debate, and think about the practicalities of delivery and what we can learn from other diseases and what's gone on in other countries. And the third session is to consider the implications for care and support. So we've got a really fabulous faculty today with clinicians from psychiatry, neurology, psychology and neuroradiology here at UCL, from Europe and from the States. We've got the chief executive officers of both of the major Alzheimer charities, experts in MS, and from representatives from our patients as well. We're going to have a series of quite short talks and then some discussions. So we're not going to take questions after the talks, but we'll take all the questions at the end as part of our discussions, and we'd really encourage people to contribute. There'll be roving microphones. Please ask those questions. There's no fire drill, so for fire alarm, then make your way to the back. Please make sure your phones are off. I'd like to take this opportunity also to thank Sandra and Ellie from Institute of Neurology and Adam from Dementia Research Centre, who set up the live stream. We've got a lot of people who are listening online. This is being recorded, and all the speakers and participants have agreed for this to be recorded. There's going to be a coffee break and drinks afterwards, which we hope you'll join for some networking. So without further ado, I'd like to introduce my great friend and colleague, someone who doesn't need any introductions, who's Nick Fox, who's a professor at UCL, who's an internationally recognised name in Alzheimer's disease, but has been involved in Alzheimer trials for more than 20 years, I'm afraid to say, Nick. So will tell us a little bit about these trials. So thank you very much.
Thank you, John.
And Nick, unfortunately, is an Arsenal fan. This rather hurts having run your yellow cards here at the moment. But I will be waving these.
Excellent. So some we prepared earlier. So it's a great pleasure. Can you all hear me all right? Great, thank you very much. Well, thank you for that introduction, John. It is, I think, a very exciting time, and it's been a long time coming. And my job is to a little bit set the scene, and I hope to set up some of the panel discussions and audience involvement that we very much look forward to. It's as much to stimulate discussion, so please do feel that there are lots of unanswered questions, and we need you to contribute to that, to our panels. So first, in terms of, I should start my disclosures, I've provided advice to a number of companies over the years, but the other disclosure I should make is that I see patients in clinic every week with Alzheimer's disease, and I have a long-standing interest in familial Alzheimer's disease, and these are the most motivating of families if you ever want to believe that there is a need for disease modification. And that is a bias, but I also, so I very much want therapies and therapies that work, but I also want to make sure that the therapies that we are delivering are really providing a benefit and changing the course for patients. And so that's really important. I also should acknowledge my funders that you see there, but most importantly, the families and patients who so generously contribute to research. So it's been a long time coming, hasn't it? There was an excitement. 30 years ago, we had the first therapies for Alzheimer's disease, and these were symptomatic, and we thought this would unleash a whole series of open the door to other therapies that would actually slow disease rather than be symptomatic. We had the cholinesterase inhibitors, and then we have memantine, and I think it's fair to say that while we may change people's functional course, they do not affect the underlying disease progression at the level of the brain and cells. And that has been the holy grail, or the real endeavor of the last 20 years, to try and alter disease progression, to try and alter the future of people with Alzheimer's disease. And now we think we may be on the cusp of that. So we've had, just so that we're all on the same page, we now have 2 FDA approvals, first with Aducanumab a couple of years ago, and only a few weeks ago for Lecanemab. And we've got great excitement in the press, but we've also got lots of concerns, and we will try and cover some of those things here. So I'll be trying to look at some of these disease-modifying therapies and talk a bit about what the results we have so far have shown, but also what some of the unanswered questions will be. So it is worth taking a step back on this long journey that we've been on, just on anti-amyloid therapies, and there are many other approaches. So this is the focus for today, and the reason for that focus today is because that's what's most likely to be coming to the NHS in the near future. But we hope this will be the first of many other approaches and many other therapies. It is over 20 years since Dale Schenk and colleagues showed in a mouse model that vaccination of these mice against amyloid dramatically removed their progression of their amyloid plaques within their brain, and then could even prevent them forming. And this led to this very hopeful cover of Nature that we would be purging the brain of plaques. And that was 20 years ago. So I think we have been taking plaques out of the brain, but we have not shown clinical benefit until now, really. So what is that 20 years journey of promise that we've been going through? It first started with active vaccination, which was a follow-on from what happened in the mice, and that is something that many of us here feel a strong association with, because we conducted at UCL worldwide image analysis for that first study, and were part of the design of it. And this was stopped early, so the trial didn't complete its length, and it also stopped treatment, and that was due to what was called then meningoencephalitis. But autopsy studies did show that there was amyloid removal from the brains of patients. We then went on to a whole lot of monoclonal antibodies and also some active vaccination studies, and I only show a few here, trying to build on the removal of amyloid to try and change the pathology and the course of pathology while reducing the risk of side effects. And now we have As I mentioned, Aducanumab has got FDA approval on the accelerated approval scheme, as has Lecanemab. And we are due we expect in the second quarter of this year to have the readout from the phase 3 study of Donanemab, which showed remarkable clearing of amyloid in its phase 2 study. So a bit like buses now, we've got several coming along at the same time, and hopefully there'll be many, many more. So what did the Lecanemab study show? And before I talk about that, I think it's worth acknowledging the role of patients and families in clinical trials and in discovery. So Lecanemab was based on observations that were first made in familial Alzheimer's disease in what is called the Arctic mutation, a family living in northern Sweden, studied by Lars Landsfeld and colleagues in Sweden, who showed that these people had Alzheimer's disease progression. They had a causative mutation in the APP, amyloid precursor protein gene that caused an autosomal dominant form of Alzheimer's disease, but they had a slightly atypical pathology, not the typical amyloid neuritic plaques, cord plaques. And they saw within these families the presence of these soluble, aggregated, what were termed protofibrils. And Lecanemab, also previously known as BAN2401, then came up, incorporated this. And so this is an anti-amyloid monoclonal antibody with high affinity, not exclusive, but high affinity for these soluble protofibrils. And that's been a major change in how we think about the disease and the course of therapies. So what did it involve? Well, it certainly wasn't for the faint-hearted, and we will hear more about what it's like to be in trials. And Lecanemab involved fortnightly infusions, IV infusions, for 18 months, as well as lots of imaging and lots of other tests. So this was a large study of almost a couple thousand of people, and what did it show? It showed a remarkable reduction in the level of amyloid. So in the yellow line there you see the dramatic fall over 18 months. And the scale on the y-axis is the centiloid scale, and at baseline both placebo and Lecanemab, as you might expect in a randomized controlled trial, had a similar level of amyloid binding at about 78, 80 centiloids. But by 18 months, the mean amyloid level in the Lecanemab group had fallen by 55 centiloids. Now these are very abstract numbers. The really important take-home message is that the majority of the people at the end of the study would not have been eligible to go into the study in the first place. The study required eligibility requirement was that people were amyloid positive, either on CSF or amyloid PET. And the amyloid PET results of the people at the end of the 18 months meant that more than half the people would not have been considered to have enough amyloid in their brains to be eligible for the study. So Lecanemab cleared amyloid from the brain. That's been shown, as I said, for over 20 years. Many of these other studies that I showed, the monoclonal antibodies, have also removed amyloid. But what would have been lacking, and we can discuss that as to why studies have removed amyloid but not shown clinical benefit until now. What Lecanemab did show in the New England Journal article that we see taken from this is it also showed on its primary outcome measure, which was pre-specified, that there was a slowing on a cognitive measure, the CDR, some of the boxes, or the Clinical Dementia Rating Scale, some of the boxes. These, again, you see the decline in the placebo group in blue, the Lecanemab in yellow, and that equates to about a 27% slowing of that decline. It also hit all its secondary endpoints, with a 26% decline on the ADAS-Cog, 24% on the ADCOMS, which are composite. And on the activities of daily living score, declined by 37%. And perhaps particularly relevant to our care and support session at the end, we also saw in this study 38% decline on what we know is so important, which is the burden on families, on study partners, and the impact on quality of life. So we've got less decline on both those measures too. What about safety? Well, these are very biologically active therapies, all of them. And ARIA-E is a feature of all the monoclonal anti-amyloid therapies, as I'm sure people are aware. About 1 in 8 individuals in the Lecanemab study developed ARIA-E. There are two forms. ARIA-E is what I'm going to concentrate on. The other, ARIA-H, is microhemorrhages. We can talk about that. And a very small level of spontaneous detection in the placebo group. And that may, we can discuss that. But importantly, there was no increased mortality during the double-blind phase. But there have been concerns raised about people after the double-blind in terms of a number of cases of cerebral hemorrhage in these open-label phases or in people who've been in the therapy. And this is one such case report with multiple cerebral hemorrhages. And this was in somebody who was receiving thrombolysis for a stroke. So we have the combination of both monoclonal antibodies against amyloid and a thrombolytic treatment. And the hemorrhages which we've seen so far have been in people who've also had anticoagulants or thrombolytic therapies. And clearly that's important for counseling patients and for cautions and how we will prescribe these therapies. So how important is this? How clinically meaningful is this? What we've seen is that there have been different views. And this is an area of active debate, but we will be interesting to review how those decisions about what's clinically meaningful are considered. And so let's just go think about this for a moment, because this is very relevant to the questions about what do we still need to know. So the absolute amount of difference, which is often talked about its clinically meaningful difference, it's maybe seen as a change in your primary outcome, the CDR sum of the boxes, at a particular stage. And so this is just puts that into numbers. So you got a certain amount of decline. It equates to an absolute difference between the placebo and treatment arms. In this case, just 0.45 points on the CDR sum of boxes. But the other way of looking at it is that these curves have been shifted, or people's course has been delayed, in this case, by just over 5 months. And that may be more meaningful because that applies throughout the disease course, whereas an absolute level of change is different in a nonlinear scale. And these are nonlinear scales. The disease is nonlinear. The disease accelerates, and these scales have ceiling and floor effects and are very nonlinear in between. And the same amount of delay, in this case I've just modeled it, for a few months delay may give a very different Y. The same X, depending upon where you are on the curve. And so you have to take that change in that context. But maybe what really matters is the amount of benefit integrated over time and the stage you're at. Maybe it's this area. This is akin in a way to QALYs, or quality of life adjusted measures. Maybe we need to create a new CALI, which is a cognitive function adjusted life years, and at the point at when that's most valuable. So what have we learned? It is possible to remove amyloid in a dramatic fashion in just 18 months. And we can reduce clinical decline. So does that benefit that we've seen require amyloid to be reduced below a certain level and for long enough? And I think the evidence is coming out now that that may be critical to why other therapies did not show an effect. Even though they removed amyloid, they didn't move enough, and they maybe didn't remove it fast enough. And an amazing thing that we just need to reflect on for a moment. It was possible to do this in people who were already symptomatic. Many people thought that it would be too late for an anti-amyloid therapy. People that have amyloid in their brains for 20 years by the time they had this therapy, on average. So this is a remarkable achievement. But we've also learned that these therapies come with risk and not inconsiderable burden. The questions remain that we will be talking about in the panel, I hope. Is the benefit sustained? This is a disease modification. It should be. Will they be progressive, additive? Will those curves diverge, or will they come together? And that is actually much the more important question than arguing about whether this 18-month result is clinically meaningful. So, will benefits be greater if we treat earlier in disease? Many of us would think that must be the case, but it needs to be shown. And might we be able to get away with lower dose, less burdensome, lower dose frequency, for example, if we go earlier? And might it also be by going earlier or by a low dose that we can make it safer as well as less burdensome? Might treatments be paused? Might we see what we've seen in other diseases, where we have a remission phase and then surveillance? What would the stopping criteria be? And if you had seen benefit in the person you love most, or thought you've seen that, would you want people to say they've now reached a level where we need to stop that therapy? What would you feel about that? What about risk? How will we manage it? Will the risk of hemorrhage reduce over time? Might it increase? If we stop treatment, might vessels remodel? Can we predict those who will respond best? Can we better predict ARIA? These are pressing questions now, and we should discuss them. And how can we deliver therapies with less burden and less cost? And we'll be talking about that later. And what about the equity and equality issues of access? So in summary, we're on the era of disease modification. We are offering hope, and that is a great place to be. And there are also questions and challenges for research, for practice, for care and support, and there are ethical and equity issues. But for my mind, I think it's surely better to have those challenges and those questions rather than no prospects. Thank you.
Thank you very much indeed, Nick. Also for sticking so beautifully to time. So please have your think of your questions for our group session towards the end. It's now my great pleasure to introduce Frederik Barkhof, who is going to talk us about some research and trial implications. Frederik is a professor of neuroradiology both here and in Amsterdam, who's been involved in Alzheimer's and Alzheimer clinical trials for many, many years. But it's a toss-up whether Frederik is more known for his research in Alzheimer's disease or multiple sclerosis, in which he's equally eminent, and has seen many changes over that period as well. So no one better placed to talk about some of these questions than Frederik. So thank you.
Thank you, John, for the kind introduction, and thanks, Nick, also for inviting me here to speak to you on this important topic. And I've been asked to talk about the implications for research and trials. Like Nick, I'd like to start off with some disclosures. I've been consulting to the various companies as steering committee or DSMB member in the Alzheimer's space, but also in the MS space. So some of this work relates to drug development for MS. And I've been leading a big consortium called AMIPET, focusing on amyloid PET, so I may be slightly biased towards PET, but I'll certainly discuss alternative options to measure amyloid pathology as well. So, yeah, there's lots of things to discuss, as Nick said, and that's the good thing about good research. It brings new questions. Negative trials bring out a lot of interesting new questions as well, but positive trials even more. So I'd like to take a step back from where Nick was and focus a little bit on the ATM framework, which has helped define the disease better and also helped rationalize the targets that we should look at. Then the question is, how do you measure them? What are the thresholds? And now that we've seen these positive results in MCI and mild AD, should we think of having even earlier treatment? Or perhaps later treatment, or, well, let's talk about stratification. And then perhaps a bit of an aside, but relevant to us, we have lots of people who participate in research studies where ApoE has been determined, where amyloid has been measured, and now these people know these results or may want to know these results. And what do we do with people who screen fail from trials, people who have too many microbleeds or don't have amyloid? What can we offer them? And then finally, what is the future of placebo-controlled trials and how do we maintain equipoise? So very briefly, this schematic from Cliff Jack and the NIA research group who helped to define Alzheimer's in a more biological manner by looking at the various elements of pathology in terms of amyloid pathology, tau pathology, and neurodegeneration, hence the ATN staging work, which is a more biological definition of the disease. And in fact, this cartoon helps us to realize that all combinations are possible. So some people have just amyloid, so only have Alzheimer's pathology. Some people have amyloid and tau, so you could say that they have Alzheimer's disease, you know, what a pathologist would call Alzheimer's disease, and they have people who have concomitant cognitive impairment or dementia, and then you have Alzheimer's dementia. So you have to be increasingly careful how we use these words. And also how we measure them. How do you measure the amyloid status? How do you measure the tau status? How do you measure the N? And so various things have been proposed, and initially it was perhaps slightly naively thought that you could use all this sort of information interchangeably, but we're learning more and more that it's probably not exactly the same. For example, for amyloid, you can measure it in CSF, or perhaps even in plasma, with PET. Tau you can measure with tau PET or with phospho-tau, but there's another tau that you can measure in the CSF, which is the total tau, probably closer to neurodegeneration, but it all depends very much on what assay you use, how you operationalize it, and in what population you test it, what is the value. One thing that is appealing to me as a radiologist, but I guess to many people, is amyloid PET, or PET in general, because it gives you a picture of the brain, and it allows you to visualize it rather than seeing some extract value and then having to know a cutoff value and then determining whether it's abnormal or not, because here you can eyeball the images. And every tracer comes with a different scaling, but there's a little trick that you can do, which is called the centiloid scaling, which brings them on a scale from between a 0 and 100 in principle, going from a normal scan where you just see some uptake in the white matter but not in the cortex to a completely abnormal scan. And from neuropathology comparisons, we know that if you have a score between 10, it's probably completely normal. If you have a score of 20, you probably have some plaque density. If you have a score of about 50, you're very likely to have an AD diagnosis at post-mortem. And then, so a visual human person can, roughly around 25, will start to call a scan positive. But there are people in a gray zone which are equivocal. And those may be very interesting for what we call secondary prevention. So we know something is happening already, but it's not full of amyloid yet, and that may be a good moment to try to stop the process and see if people may not develop symptoms. And as Nick said, it probably depends very much on, with the amyloid lowering therapies, where you bring these subjects. The positive studies all were able to get close to the 20 centiloid level. And the ones that failed, for example, Aducanumab sadly failed, reached this level, so had a very significant reduction, but didn't bring it to the 20-25 mark. And even for the two Aducanumab studies, the one that failed had less reduction than the one that was positive. So it's probably critically important how much amyloid you remove. So let's talk about how to measure it. So PET is one thing, but as I said, you can measure the CSF and perhaps in plasma. Well, the plasma markers will be preferable, of course, but the tests are not good enough at the moment. CSF is probably very close, but it's cumbersome, but you know, PET scan is also cumbersome, you could argue, so that's a bit on par. And it's actually very interesting, if you start looking at the relationship between PET and amyloid levels, they're overall related in this sort of hyperbolic fashion, but there are some people that sit on this side of the curve, and there are some people that sit on this side of the curve. So some people have a PET over CSF imbalance and otherwise a CSF over PET imbalance. And if you look at the rates of cognitive decline, they differ between these groups. Probably because the CSF positive group is a bit earlier than the PET one, so you have more lead time. But that is something that we need to study, and also indicates that you can't just use these interchangeably in clinical practice either. So what does this mean if you want to do a secondary prevention study, such as the AHEAD study, which tries to look at Lecanemab given to people who got screened out of the population with either just a positive scan or a sort of borderline positive scan. And they used ApoE and plasma tau to look if they could predict a positive scan. This is a slide that got from Risa Sperling, who's leading the AHEAD study. And they showed that it's actually reasonably achievable, especially the p-tau 217 does a pretty good job in predicting it. And if you then use a specific amyloid assay, mass spectrometry assay, and you combine them, it's even better. And the idea for this study was to get 100 amyloid positive people, amyloid positive scans in, and you would need to have screened 3,500, and with the help of these assays you may reduce that to less than half or almost a third. So that's a significant saving in costs and burden to people to do that. Or perhaps people want to self-screen. There is something called euphemistically brain health services, which is an emerging trend where people who are, sometimes referred to as worried well, self-report to the doctor, say, well, can you test my amyloid? I want to know whether I have amyloid, and perhaps see if I can seek some treatment for it. Or other interventions. Giovanni Frisoni and a group of people have recently published this paper describing, what you can do in terms of risk assessments, but also some lifestyle advices, and perhaps at some stage also offering monoclonal antibodies in appropriate settings. So this is something to watch out for. And then finally, obviously, keeping in mind, what the well-being and the insurance issues around having, finding out that you are amyloid positive, for example, or carry the ApoE4 gene, because obviously that could also be a burden in itself. And then finally, at some stage, you could daydream about population screening, but then we need lots more data about the benefit-risk ratio over longer terms in wider populations, comorbidity, cost-effectiveness, which will be difficult to prove. And I guess probably you would need to do some better risk stratification by combining not just amyloid but also tau markers, see where people are relative to real risk of developing cognitive decline. And that is the big question, have been the big question all along, I think. Are we too late or are we too early? I think the earliest studies looking at amyloid antibodies were probably too late. They included moderate to severe AD with a lot of neurodegeneration. And if you've lost a lot of neurons, who bothers to remove the amyloid, because you're not going to regrow your hippocampus. So most studies have now moved to early AD or MCI, and some of them even limit the amount of tau pathology. In other studies, it's unknown how much tau pathology there is. Or should we, as alluded to, should we go to asymptomatic people who we screen out of the population who are amyloid positive? But these are not easy studies to do. You need a very long follow-up to determine whether there will be, in the long run, a cognitive benefit for these people, to determine the cost effectiveness. And you might argue that you need to be probably relatively close to the onset of tau pathology if you want to have a meaningful impact. My colleague Rick Koppler looked at this, collated a large number of data sets from people who were cognitively unimpaired and were either completely negative, had only amyloid, had amyloid plus a little bit of tau, but only in the medial temporal region or in the whole cortex. And it turns out that those without tau, they are very unlikely to decline much over time. So this is progression to MCI, this is progression to dementia. Whereas if you had some level of tau, then at least the risk for MCI was substantial. And it's good to realize that some studies have tried to limit the inclusion only to this population, thinking that this is already too late. You're too much towards the dementia onset already, or in the dementia phase. And that's probably a sort of realistic choice to try to be somewhere either here or between here and here. Similarly, as we saw for amyloid, how do you measure tau pathology? You can do it with PET, you can do it with CSF, probably best with p-tau. And here again, people have looked at what is your risk of survival. And survival was here clinical progression to MCI or dementia. And you can see that if you were positive on both markers, you had a really big risk of decline. But some people that were discordant also had a risk. So if you had either of those markers, you already had a risk. But it's not as much as when both markers were positive. So I think we need to learn more about the discrepant values and what that means. So what are the implications for research studies? This is perhaps a slightly tangential topic, but there are many long studies ongoing, like the 1946 here at Queen Square, the amyloid PATH studies that I was involved in, where typically we do not disclose the amyloid status, but people we know that 20 to 30% of them have amyloid also, if people select randomly. And they now may come back to us and say, well, why don't you tell me whether I have amyloid? Because I've heard there's a drug for it. What do you do with that? Not to mention what happened to these studies, but that's a secondary point. Because we know that the amyloid starts probably 20 years before you have dementia. So what do you tell these people? Not an easy one. Obviously, the studies will be endangered, but that is something that we need to solve. And they could also be plagued by people who want to come into a study because that's a cheap way to get an amyloid test because I can't get it in from my GP, or they may go to a paid brain health service in the first place. Another thing to consider is what do we do with all the screen failures in trials? I don't think whether Nick mentioned that, but there's a long list of in- and exclusion criteria that were employed for trial recruitment. And typically between 17 and 90% of people did not go into the trial. The majority because they didn't have amyloid on whatever measure, but then for a whole series of other reasons, including having microbleeds. But there's lots of other things why people couldn't go in. So we need to think, if this drug is available, do we want to be as strict as that? Or what do we do with people with risk factors who have anticoagulants, for example, or have microbleeds, or both? Do we disclose this, and what does that mean for people to learn all these laboratory tests? What do they do with it? Around microbleeds in general, there's a lot of discussion, of course. How many can you tolerate before you go into a trial? How many can you tolerate once you're on a drug before you should stop the drug? What about people needing anticoagulation? Is there another threshold? And hopefully there will be alternative treatment options for these patients. For example, the tau, anti-tau studies are now also showing some stuff. And then there are people, if you remember the cartoon initially, who have neurodegeneration but no amyloid and no tau, euphemistically or descriptively referred to as SNAPs, suspected non-Alzheimer pathology. Well, you have an issue, but we don't know what. Some of them have TDP3 pathology. It would be great to have a treatment option for those people, but also to have a means to recognize that in vivo. And then finally, I'd like to finish off with this slide about future randomized controlled trials. One of the principles of RCTs is that you need to have equipoise, which means that there's always a balance with reference to the standard of care. So if one of these drugs would become the standard of care, should we then include an active comparator arm? I guess so, if we think this should be the standard of care. But as you've heard, the long-term effects of anti-amyloid treatment are uncertain, so you can have long debates about this. One way forward could be to do head-to-head studies. For example, the run anti-amyloid versus anti-tau, or versus a TRAM antibody, or another treatment. Or you could do combination studies. For example, say, I want to do amyloid removal plus or minus a tau antibody, or stratify the groups depending on whether they have concomitant tau pathology. So in the MS field, we've seen that in the current days, there's no equipoise anymore to do a placebo-controlled trial. So you need to have an active comparator drug. So what you see now, we have 15 registered drugs, and we'll hear more about it later. So people tend to use the least active comparator as a reference group, but then at least people are given something, and then you can perhaps show a benefit. And all of this may depend on the disease phase in the population. If you're in a primary or secondary prevention study, you may have a different stance than when people already have MCI or mild dementia. So, as you've seen, I have more questions than answers, but I think that is what the whole afternoon is about. So I look forward to discussing all of this with you. Thank you.
At this stage, I'll going to invite Frederik to come and stay up here, and we'll ask Nick to come up as well, and Catherine Mummery as well, and also Hilary Evans, who I think is coming up here as well to come and have a seat. So just to introduce anyone, Kath Mummery is co-organising the course today. Will be chairing the next session, and Kath's the clinical lead for dementia and therapeutics here at the Dementia Research Centre, with a lot of experience in running these clinical trials. And Hilary, as well as being the Chief Executive Officer for Alzheimer's Research UK, has recently been appointed as the co-chair of the UK's government's dementia mission. So it's a great pleasure to have everyone here today. So we're going to have roving microphones, so let's identify any questions. Before we get started, I thought we might just turn to Hilary with your new role, not least as Hilary has had an opinion piece on the front page of The Guardian today about these treatments. So Hilary, just to reflect on, from your perspective with your governmental role and your AR UK role, about where we're at with all of this.
Sure. Thank you, John, and real pleasure to be here today. It's a fantastic symposium to be a part of, and I've nicely timed the front page of The Guardian on dementia research and the hopes that we have and that I have in this space. And I think one of the conversations I had with the journalist who wrote this article, Hannah Devlin, who's the science editor of The Guardian, was actually her reflection as she has spoken to people in this field in recent months, was actually none of you are confident enough that you're going to do this, and I don't get that in other areas of health. And I've been a reporter for a number of years, and I speak to people in cancer and heart disease, and there's excitement around new drugs coming through, there's excitement around what's happening in clinical space. There's a real sense of momentum, but actually she was saying, I can see that that's all here. And I want to talk to someone where we've got that excitement and we can portray that. So hopefully I've managed to do that in that piece. And I guess, from where we're sat, from Alzheimer's Research UK, from the kind of wider community, I do think that this has to be a turning point. My role as co-chair of the Dementia Mission is really to advocate for patients. And this is something that, as an organisation, we have campaigned for in recent years. So we saw the role that Kate Bingham took on in COVID, championing the UK as a place to do COVID clinical trials to ensure that UK citizens were some of the first to get COVID vaccines. And so really we're trying to take some of the learnings there when it comes to dementia, and really just to try and speed up and hold government to account in this space as well. So this role comes with increased funding for dementia research, but also a brief really to me and to my co-chair, Nadeem Sawar, who works in industry, works at ESAG, really to tell the government what is needed in this space, to work with all the different bodies to try and join up the infrastructure. So from where we're sitting, we can see the very, very clear need. We all know a lot of the statistics, leading cause of death in the UK. What is it that we are doing? Where is that sense of urgency? There is huge progress being made. We can see that through the presentations and the conversations that we will have this afternoon. But I think Nick set that out really clearly, the progress that's been made in 20 years. And I think that's really, really exciting. But I do think the UK, it risks falling behind because we aren't deemed a brilliant place to do some of these clinical trials. We don't have anywhere near the sort of infrastructure to ensure that patients gain access to these new treatments as they come through. And so for me, that's going to be a big part of my mission role. And I think that with that COVID experience, we've seen that interest in medical research. We've seen an interest in people wanting to take part in research, clinical trials, more than we've ever seen before. So some of the work that we've done at Alzheimer's Research UK in our Dementia Attitudes Monitor shows that 69% of the population would be keen to take part in a dementia trial. So we need to ensure that we actually have trials in the UK that they can take part in. And I think that obviously brings huge benefits for the patients. It brings huge benefits for our clinical community in terms of getting an understanding of what those treatments look like. But also it helps us make those cases to regulators, to NICE, and to the NHS in terms of how we can prepare for those treatments. So there are sort of threefold goals, I guess, for the Dementia Mission, and we are working with those across the sector. So obviously there are lots of different players here in the UK and globally working in this space, and the mission is really a brief to sort of join some of that up, but really look at threefold in terms of how we can do that. So we have a brief to look at sort of biomarkers in terms of how we can ensure there's a good, accurate diagnosis for people developing these diseases. We can look at the wider clinical trials infrastructure, and that's something that we're doing alongside Kath in her new role, and she's going to be talking about that today. And then to really champion the needs and wants of our patient community in this space as well, and to then join up some of the other initiatives in this space so that we do work smarter, that we can work more effectively, and just keep up that pace and that momentum that I think we need to see.
Great. Thank you very much indeed. So if there's any questions, then please put your hands up, and we'll have our microphones come round. But I mean, we'll kick off the discussion. I think a very real question that Hilary raises, and perhaps we'll come to Kath for this to start with, is around recruitment to clinical trials. You've done spent a lot of time recruiting people. Do you think that these sort of successes are going to make it more or less difficult to recruit to clinical trials, and particularly if we're going to have therapies that are out there? Do we increase the number of people who are interested, or is this actually going to make things more difficult when it comes to recruiting to clinical trials?
I think that's a really interesting question, because I don't think there's a simple answer to it. At the moment, it's increasing interest dramatically. Whenever there is a news story, front page of The Guardian, we'll have 150 people tomorrow wanting to go into clinical trials because of the story, which is brilliant. Whenever there is a phase 1 study or a result that comes out, you have direct contact from people.
Specific or generally, and I will think of somebody.
Well, I have a question, I think, largely for the panel, probably for Nick and for you as well. Can you hear me?
Speak up a little bit, if that's okay. Sorry, yeah.
Is that a bit better?
That's better,
yeah.
Thank you.
So, this is kind of a
sorry, could everyone ask a question? Could you just say who you are, just so we, yeah.
Right. I'm Gina Swartz. I'm currently Chief Medical Officer of a company called Exiva. I was recently Global Clinical Head of Neuroscience Drug Development at Merck, or MSD. So I have a multifaceted question, forgive me. But really, when we look at the current evidence coming out of the monoclonal antibody trials, you've indicated that we need to be able to reduce the level of amyloid as depicted in PET or potentially CSF by a significant amount. Now, when I look at the failed studies, and I look at, for example, BACE inhibitors, where we did very much the same, and for example, with Verabesistat, we were able to eliminate amyloid by a degree of 90%, and we actually showed a worsening in cognition rather than a benefit. And the consensus thereafter was actually that our doses were too high. The question I have here is, how do we then reconcile what we've seen previously and what we are potentially seeing now with the novel emerging therapies? How do you then put this into a clinical framework whereby you are defining the baseline level of amyloid and then looking at the degree of reduction. Do you then do this in a methodological way in order to stratify patients for clinical inclusion in clinical trials, or ultimately those who will benefit in the clinical paradigm? And then how is that best done? So I know that's a lot of questions and I'm not expecting an answer, but I'm sitting here feeling somewhat confused about why the paradigm we're about to embark upon is so different. And then the next question comes about safety as well.
So if we sort of distill this down, these are joint questions about the level that you've got to start with, that clearly amyloid removal per se might not be sufficient. So how can we reconcile that? Nick, do you want to come in on that?
Thanks. Really interesting question. So the BACE inhibitors reduced amyloid production. They didn't clear amyloid from the brain in the same way. And I think these are different mechanisms. And what we have learned with the BACE inhibitors is that altering the amount of amyloid beta monomer is probably not the same as taking out protofibrils. And so I think that's a... We've learned by our mistakes, haven't we? Yeah. You asked how will we reconcile and how will we know. One really, it sounds a politician dodging the question type of thing is, well, the next trials will tell us. Because if we see this as a class effect, if Donanemab produces this, if the open label extension show, then we will see, I think, that the evidence is coming together that if you remove enough amyloid, and I'm using that very loosely, all the different moieties along that cascade, low enough for long enough, we will get benefit. I think we have moved the point beyond the point where people could debate, oh, there's no point removing amyloid, it'll be too late, or we don't know whether that's going to help. I think that the old story of the bullets out of the gun, the triggers being pulled, has been shown, at least in at least one study now, to be wrong. And that is great for the field. I probably didn't answer half of your question, but that's one of them.
Frederik, do you want to come?
Yeah, can I add to this? So I agree, it depends really much on what type, because amyloid comes in multiple forms, monomers, oligomers, and fibrillary, and I think just reducing the number of monomers is not enough, and some antibodies, for example, Aducanumab failed because it was too much towards the monomer target, and didn't do enough on the fibrillary amyloid, and the ones that are have an effect all have the side effect of ARIA, and we'll talk about ARIA in a minute, and it's a side effect that we want to avoid, but to a certain extent it also means that you're engaging with the tissue and you're removing the amyloid. So that's a sort of positive effect of this side effect, that you at least know that you're hitting the target. And now we probably need to find a drug that does both removal of the fibrillary without introducing too much ARIA.
Yeah.
Just to add to that, I mean, Frederik, you made the point in your talk. If you remove low enough and long enough, it makes a significant difference, and there is a margin. So if you look at even the same studies you said, Engage and Emerge, one was positive, one was negative. One removed above 25 centiloids, one removed lower than 25 centiloids, one was positive, one was negative. All of the studies are consistent. If you look at how much you remove by, you see consistency in terms of the effects. So I think the failed trials and the positive trials are now giving us a converging set of evidence that helps us understand the differences that you're talking about. And I think, as Nick said, that has moved things now, so that regardless of what the trial is next, whether it's positive or negative, the information will really help push that forwards and give us increasing convergence. So I think that's a very different position from where we were at the BACE inhibitors, where we didn't know anything.
No, and a final point, I think we need to better understand the population that is being treated. There's lots of heterogeneity within these trials. I know that for a fact. I've looked at thousands of scans of people who goes into trial, and some have beautiful scan, and others are completely atrophic, and they all go in under the label of MCI. So we need to find better. I think the first studies with Aducanumab didn't use amyloid as a pre-screen. Turned out later that probably around a third didn't have amyloid in the first place. We know better now. Some studies now use tau PET to titrate the amount of tau pathology that is allowed to go in. And so we need to progressively better understand the population that is going into this trial and stratify patients better based on a variety of measures that we have to understand who will be favorable responders and who won't.
I think that's also a very important point, because I take a step back and say, look, where do we want to be? And I think the best analogy for this is probably cancer trials, when you're almost having a bespoke treatment for each individual. We're at the moment got this morass of a placebo-controlled trial, and what we're going to need to do is to move from that to a personalised medicine approach that will be on genotype, and we'll discuss the issues around ApoE genotyping later on for your risk for ARIA, then the right drug at the right stage. But at the moment, we've got an extremely exciting first pass with a positive trial, and then we need to work out the intricacies between that and getting down to the individual patient level. So perhaps I will then again ask a difficult question to Hilary, which is one of the issues to be able to do this is we need access to all of these data. And how can we incentivize, say, the big pharma companies to make all the data that we have open access? Not that I'm posing you as my boss at AOUK with a challenge, but how we can, in your new role, is that we can make this as open and transparent as possible. Because if we're going to understand this, it seems to me that we need to have as much access as we can, as soon as we can, to the raw data to be able to understand that. I don't know if you have any thoughts on that or how we might do that.
Thanks, John.
Sorry.
I wish I had the answer as how we might do it. I think, I guess a few responses on that. So I think data is something that flows through a lot of what we want to look at as part of the mission. I think that the mission gives us a unique perspective to kind of sit across all the different initiatives in this space, funders, governments, and champion why we're all here and doing what we're doing, and maybe to use that platform to then be able to say, what is it that's going to make the difference, and open access to that data and understanding of that data and how it's used is really key. I think that wearing my CEO and charity hat, we run Joint Dementia Research, and obviously we've been championing the need for all individuals to sign up to dementia research, particularly those with MCI or a dementia diagnosis, and that's still only 2% of people enrolled in JDR. So I think that there are some big challenges, but I think actually as a charity, as a sort of trusted voice advocating for patients, is to be talking to industry and ensuring that the data then does deliver. So I do think there's going to be a role for the mission in doing that, and you'll probably hear more from us in terms of then thinking about how we do that and looking at some sort of world experts who've done it in other disease areas.
If we have any other questions, then please ask. John Roy.
Thanks, John Roy, ECL. Kath, I was just interested to know what conversations have been had already about how trials will change in terms of a new standard of normal care, and what conversations have been had about dual therapy trials and anti-amyloid plus tau. You're obviously doing trials in multiple other things that are non-amyloid. Thanks,
John. And this is the topic of choice when a company starts talking about what they're going to do with their phase 1 trial, their phase 2 trial. The window of opportunity people talk about, because drugs aren't here yet. It's a very interesting space. People don't really know how to manage it at this point in time. Lots of discussions around the ethics. There's one position which is quite extreme, what you have taken, which is that it's not ethical anymore to have a placebo-only trial, and they've already started combination trials. So one of the trials we're doing in people at risk of genetic Alzheimer's disease, they start on open-label Lecanemab every 2 weeks for 6 months. The theory being they clear out the amyloid over 6 months, and they then either go onto placebo and lecanemab, or they go onto tau antibody and lecanemab, which is a massive addition of complexity to a trial. And it's been hard enough to get to positivity in just anti-amyloid immunotherapies after 20 years. So that adds a lot in terms of complexity of analysis. It adds a lot in terms of the burden, because they're coming bi-weekly for those treatments, and then they add on the second treatment. And it adds complexity in terms of safety. Personally, I think they jumped the fence a bit quick. I think there is still room to be doing placebo trials. However, the conversations now are about whether you do those trials in countries where they can't have that drug yet. And that's an odd ethical discussion to have. I haven't got a solution to that, but I think over the next 18 months what you'll find is there are increasing conversations about how people are going to design in the future. And I think you'll find either combination therapies in Washu's style in about 2 years' time, or you'll find the sort of Reese Sperling has gone a different way, and she's got, I think, 6 different arms to the next study that she's designing. So there is a placebo in there, but there's also combination therapies and all sorts, and that again will be so difficult to understand. So it's a very unknown space, and I'd be interested in the MS thinking on this at that time, because we're in that odd space between placebo and no placebo.
Very, very short comment. So, about the ethics of doing placebo-controlled trials in other parts of the world has been, and probably still is, popular in the MS space. And I think it's, if you think of equipoise, you have to consider the local situation. If there's no drug available, then being randomized between placebo and active drug is probably acceptable, even though we would think, in our circumstances, it wouldn't be, and it's been quite popular for decades.
The only comment I would make is that at the moment the indication only is for MCI due to AD or mild AD. So I think it's likely you will continue to see placebo-controlled trials as that window expands. But over time, I think the pressure will move towards everybody will need to get an active treatment. So people will be, as I think, Frederik, you were saying, should we be going earlier or should we be going later? I think what will happen is people will go earlier and later. Needs must.
Got a question at the back.
Hi, my name's Omer. I'm founder of Neurometry, which is a digital neuroscience tech company. We work on early detection using digital markers for MCI. Question I had is a framework comparison between North America and Europe around MCI particularly, and it's the conversation between research versus clinicians. So when we try to evaluate or recruit patients, there's no designation of MCI around GP. They seem to fall through the cracks a lot in Europe because it's not formally recognized in many of the regions. In North America, the advocacy was a lot better around MCI, so my grandmother went through a detection process in Rochester at the Mayo Clinic, and we could evaluate the pathology. Having said that, the issue first is what is being done to advocate signals of MCI within clinician or practice NHS, and secondly, in terms of regulatory framework for taking digital tools up towards the front end, basically, to the users. The process is still quite onerous. Is there any advocacy around fast-track processes, similar to how, orphan class was addressed, etc.?
So there's 2 questions here, one of which is a framework around MCI, which perhaps we can actually take in the clinical session. You're absolutely right, there is no nice guidance for a diagnosis of MCI, and the MCI diagnosis is hugely variable. For those of you who are non-clinical, this is mild cognitive impairment, so people in a pre-dementia state, and that is an issue that we come across in the UK and Europe a lot, is very different understandings of that. But I think it starts, the question really is about going earlier and recognising earlier, and then using molecular diagnostics. But that's the second question, which is more allied to the research, and I think maybe we could put to Frederik and Nick, is about how we what you're talking about is how we validate new biomarkers, I think, in a clinical state, and I guess both Nick and Frederik have got a lot of experience with imaging, but also coming to some of the fluid biomarkers. And I guess one of the questions that we have a difficulty here is, how do you know something works until you have a drug? And once you have a drug, then how does it work? I don't know, Frederik or Nick, if either of you want to answer that question.
Yeah, so it's often a long process, as you've noticed, also because the goalposts may be shifting while you're doing it. But first of all, I guess you compare your technique to whatever gold standard you choose. For example, being able to detect that somebody has dementia or somebody is amyloid positive, you can define. And then the next thing is to show that if you can, level yourself to that, are you then also be able to make some predictions towards a future development, and are there any false positives, false negatives, how does it work in a real life situation without being properly instructed and observed? And then finally, but it's a really difficult one in terms of a surrogate, can you also predict the treatment effect, or if you see a change in your digital outcome, is that then also predictive of a long-term treatment effect, which is often impossible to show. So, wishing you good luck.
Very, very little to say that, but I do believe strongly that often people have it's true for treatments across all of medicine, it's true for biomarkers across all of medicine. People have a great belief in something. And actually evidence has to trump that. And the process of gathering the evidence and really showing its role and its place, evidence behind that, is absolutely essential. We mustn't, even though we're keen to short track something we know is going to work well, we have to have the evidence.
Chris? I just have a comment from online commenting on the importance of recruiting diverse patient populations who are representative of the NHS population that will eventually receive therapy. So I don't know if Dr. Mummery wants to touch on that.
That's so important. And we look at the populations in our clinical trials, and they're 98% white, middle class and Caucasian. That's just terrible. How can you generalise from that population? So that the comment is absolutely right and valid, and we need to do much better at reaching out to communities that are traditionally underrepresented. It's very well done in South America, for example, and it's much better done in the States now than it was done, and we should learn from their examples. But we need to stop expecting people to come to our nice plush clinics in our UCL or wherever hospitals. We need to go out to communities, have appropriate community champions that understand and have the trust of their communities, and take research to them. And there are some initiatives that are doing that now that are showing that that does work. It worked with COVID, that's a very different environment, but we can learn some lessons from that. I think the other point about that, which we fail with research to do, and it's related, there was a great article in The Guardian, not Hilary's, which was fantastic, but there was another one from Andrew Pollard, who was vaccine chief at COVID, Oxford, talking about difficulties with incentivising research and getting distributed people into research. And it was about the need to value research in the NHS. If we do that properly and embed research within the NHS and our trials within the NHS properly, we will get a more diverse population. They'll still be slightly underrepresented, but they will be much better represented than those that self-screen, as Frederik was saying before. So really valid point, and an awful lot of work that we should do, but it's really important. And one of the things actually that I want to focus on as in my new role as chair of the DTRC, the Trials Network, setting that up and increasing diversity nationally in our trials.
Hilary, do you want to come in on that?
Yeah, I mean, I'd echo a lot of that, and I think it'd be something that Kath and I will want to be working on together. And I think one of the big challenges, and the many challenges of the NHS at the moment in dementia is essentially being set up in the right way to deliver for people with dementia and to deliver for people who have concerns around MCI or memory problems. And so I think there are different models that different parts of the UK are sort of looking around sort of brain health clinics and how you kind of bring people into the system. But my dream would be everyone who is going into that system should be offered the opportunity to take part in research, and that obviously will kind of broaden that base. There are some particular bits of work that we're doing as a charity because we know a number of people who are at high risk of developing diseases caused dementia will be from those hard to reach communities. So there's specific things that we are doing as a charity to raise awareness, and I think probably one of the biggest things that we've done in the last 10 years as a charity is just to go out there and talk to the public about the fact that we're talking about disease, it's not an inevitable part of aging, so that the work that we're doing in terms of the just major public awareness is getting people to understand that there is medical research in dementia, and you can start asking these questions, and I think sort of stirring up people's kind of interest in this, kind of outside of university, college, hospitals, and those who are going to be reading articles in the newspaper that we might write. And I think that that's where we're going to make the biggest difference. And so we need to be really smart about how we do that. But there's going to be a part of, I think, the work of the mission where this really does need to be a priority. And there are things that we can learn from COVID and others in terms of how you take this out to the public. But we have to get the public behind dementia, and we have to get them excited about the research in order to take part in research.
A very quick comment if I may. There's one really underrepresented, neglected group that never gets talked about, and that's people who don't have somebody to advocate for them. If you're single and elderly, you will not get to a clinic until you're very advanced, you will not get into a trial, and you'll be excluded from the trial. And that will be a major challenge for when we move, particularly as we got, hopefully, increased longevity. There will be many more people who will just get missed out.
Thank you very much. I'm Bob Barber, old age psychiatrist. I'd just be really interested in your views about how you see the role of amyloid playing out in the conversations people might have in the future about the treatment. So obviously at a group level you can see differences between the correlation between reduction in amyloid, but can that predict response for an individual, do you think? So, or another way of putting the same question, 30% of people in the Lecanemab study probably didn't fall below that level, and did they respond or not respond to treatment? It's just the power for that to predict response and how you see those conversations playing out.
Who would like to take that? Nick?
I can take a start. It's a great question. So one of the questions is as in all therapies, responders. How do we predict people who will do best? And I think we're just at the beginning of that journey. But I don't think we should completely feel that the people who didn't hit those really low levels of amyloid in that 18 months didn't have a benefit. We don't know. They may not have had any benefit. They may, quite possibly, thinking about a cascade, they may actually have a delayed benefit, or maybe they'll get benefit when they get their additional therapy. So it's a great question. The short answer is we don't know.
Possibly when you think other measures might be helpful as well, the tau and the neurofilament and so on.
Yeah, again, others people. I think there will be huge amounts of data. And there are several people in the room from various companies, and they will be sifting through those data now, trying to work out who will be the most appropriate and the most likely to respond. One thing I can tell you is that when donepezil was started, I remember, it was a bit before my time, but we were initially told that you'd start donepezil, and if people didn't respond, you would stop it. That did not happen. It just did not happen. It wasn't possible to sit in front of somebody and say, oh, well, I don't think you've responded, we're now going to take this therapy away. I think we'll have exactly the same thing, even if we know who's likely to respond better and who doesn't.
It sounds to me, there's a feeling that there's issues around safety, but then one of the biggest ongoing questions which relates to efficacy and to cost is around stopping. When do we stop? And that plays into some of those questions as well, isn't it? Is it just a matter of stopping? And we need much longer term data. So I guess one of the questions, I don't know if anyone answers to this, is how we incentivize or put in systems to follow up these people. I mean, Nick, you can speak to how much we learned from the AN1792 trials by seeing the post-mortem data, ultimately what happens to people. Catherine, if you want to talk about that. Yeah.
It's a fundamental question, isn't it? I mean, Nick's just made the point that trying to stop a treatment when there is no other treatment that's a very, very difficult, impossible conversation to have. But in terms of stopping criteria, which I think is something we do need to try and define, and we don't know right now. We now have data, though, or we will have data soon, on people that have been on drug for at least 8 years. And that information is hugely valuable. In terms of when to stop a drug, I think also it always echoes with me what participants have said, which is they want the drug to give them time, but they want it time at the right stage. There's absolutely no point giving a drug to buy somebody time when they don't feel that they've got good quality of life anymore. And I think that is a really important principle. That's in discussion with an individual at the moment because there's no evidence behind it. Monitoring what happens once these drugs come in, the phase 4 trials, post-marketing trials, will be absolutely critical. And I think one of the things we need to do here is look at some similar system to the ALZNet system, which has been set up over the past 18 months in the US, which basically every person that gets given drug is on a register, very light touch, very brief cognitive and physical tests, and they're followed to see what happens. We will have invaluable information from that on every single person on the drug. That's how we start to learn about exclusions, about lightening the criteria, about stopping.
Comorbidity.
About comorbidity, exactly.
So we've now raised many more questions than we've answered, which I think I see as an absolute benefit. The fact that we now have drugs that have a degree of efficacy that allows us to even ask these questions is a huge positive step. We're going to stop now for coffee for half an hour, and then the next session we're going to think about the better clinical implementation. So take this opportunity to give everyone a round of applause and thank our panellists for this. So please be back here promptly at 3:45. Thank you very much.
Is it this one? Welcome back, everybody. I can see the one stray we have at the moment is Professor Fox. Welcome back. Can you hear me okay? Yeah, brilliant. Okay. So that was a fantastic session. Yes?
Check that you can hear you online, because there are 100 and something people online.
Brilliant. Apparently I'm loud.
That's nothing to do with the volume.
So I hope you enjoyed your tea. That was a fantastic session, and as people have said, there were more questions that came out of that than answers, and I could have sat there for 3 hours failing to answer questions there. So maybe we need another session, John. But we've heard about possible implications of treatments now in terms of trials, but what does it mean in terms of our services? Where are we now, and where do we need to get to, and what can we learn from others? It's a really exciting time to have these treatments. It's a great problem to have, as I think Nick said earlier, but we have got quite a journey in front of us. So what I would like to do is just briefly go through the presentations we've got in this session. It's a packed session, and I'm delighted that at the very head of this session we have Rachel, who is an old friend of mine who has done trials with her husband for how many years now, Rachel? 6 or 7 years. The amount of time she's committed to our treatment trials is extraordinary. So thank you so much for being here. Rachel's going to give us a flavour of what it's actually like to have one of these treatments and be in a trial, and John's going to guide them through that discussion. So that, I'm sure, will inform the rest of the discussions we're having. I'll then update you about where we are in terms of services right now and where I think we need to get to and some of those challenges. We'll hear from John about what's happened in the States, their experience, and we'll hear from Heather, who has many more years' experience of having treatments that actually make a difference in MS. And we've given her 10 minutes to give us all the solutions to all of our problems. So I'm sure you'll be absolutely fine. And then we'll have a round table with everybody, and Nick Fox will join us to try and answer your questions. So again, please keep your questions to the end, and please have lots of them ready for us. That would be great. It would be great to have a really lovely discussion. So I'm going to say no more. I'm going to hand over to John.
Thanks very much. So there's lots of discussions about new treatments, but I think it's rare, if ever, that we actually hear from somebody who has actually been involved in any of the clinical trials. And so I'm absolutely delighted that Rachel and her partner Steve are here with us today. Before I start, I'm going to share some clinical information about Rachel and her care, which they are both very happy with, and they're aware that it's being recorded and live streamed. As Kath will say, Kath's known Rachel for a very long period of time because Kath has been in charge of the clinical trials. I'm Rachel's neurologist, and as you'll see, I've diagnosed Rachel, and I have seen her on a routine NHS basis over the last 6 or 7 years. So I'm not party to any information that's not in her clinical record, and so I can make any observations that I want without breaking the blinds of any clinical trials. So I'm going to very briefly, and again, this is Rachel and Steve have seen this, so I'm going to just show you the journey that she has been through, and then we'll go through some questions. So Rachel was referred to my cognitive disorders clinic in 2016, so now 7 years ago when she was 62. Rachel's university educated and managed social housing for a London borough before retiring at the age of 59, not because of any cognitive difficulties, but because the job changed. And your symptoms started when you were 61, and you were a bit more forgetful, more reliant on lists and calendars, losing track of conversations, and a bit of difficulty with time management, a little bit more repetitive, loss of confidence, and less confident navigating, and it was beginning to impact on your day-to-day life. And that's when you came to see me. The background history is of thyroid cancer and breast cancer, both of which are treated and in remission, and there's no family history. And when I saw Rachel first, she scored 28 out of 30 on the mini-mental state examination, losing a couple of points for orientation and recall. She had an MRI scan which showed that there was some disproportionate volume loss, including the hippocampal, so evidence for neurodegeneration. Rachel had neuropsychometry at that time, which showed a very preserved verbal IQ, but below expected on performance IQ, and evidence of problems in several different domains in terms of memory for words and faces, multitasking, and a bit of slowing. And Rachel went on to have a spinal fluid examination at that time, which I'll just take you through here. I put the actual figures here, so we measure the amyloid concentration, which unlike PET goes down when in the presence of brain amyloidosis, so that was low at 325, and the tau level was very increased, and the phospho-tau level was increased. And on that basis, I diagnosed her with mild Alzheimer's disease, and this is fulfilling all contemporary criteria, so this is the NIA criteria with biomarkers, and IWGT and all research criteria with evidence for multi-domain cognitive impairment, for progression, for evidence of amyloid and tau pathology and neurodegeneration. And she was started on donepezil. At that stage, she was recruited to the EMBARK trial of Aducanumab, now Aduhelm, and started dosing in 2016. In 2019, as you all know, the trial was terminated. But then there was a re-enrollment and open label stage in 2020, which, Rachel, you continue in now, and you're due to finish in October 2023. And as I say, without sharing any details that are outside of the clinical record, which she's happy to share, I've been seeing her on a yearly basis since that time, and you can see that the mini-mental state examinations have held up. There was a bit of a hiatus during COVID when I neglected to try and do an MMSE properly over the telephone, but we met again in 2021 and 23, and the mini-mental state examination is still 27 out of 30. So perhaps, Rachel, at this stage, if it's okay, we might just talk a little bit, and I hope you'll, about how you are now and how you feel your memory and thinking are now.
I think I'm more or less holding my own, perhaps just deteriorating, but very slowly. That's what I feel.
And the sorts of things that you...
Witness my
yeah,
job.
well, I'll hear from you and then see what... And the sorts of things that you're enjoying doing?
I'm still cooking. I'm knitting. My baby, most recently, the cardigan. I play table tennis every day. That is dry. Outdoors, and I'm still cooking.
Scrabble.
Oh yes, I can win at Scrabble.
Is that because you're good at Scrabble? Stephen's not so good at Scrabble. I won't ask that.
Rachel usually wins at Scrabble.
And in terms of sort of the things that you're enjoying doing, you've got new grandchildren,
Yes,
that's right?
I've got two grandsons.
And what are their names?
James and Frank.
Good. And how old are they now?
Well, Frank's the newest one. He's just weeks old. But James is 3, I think, isn't he?
And you're playing quite an active part in their lives as well?
Yes, although sadly James lives in Bristol.
Okay.
But also it's easier to see the other one.
Yeah, but can everyone hear, by the way? Yeah. But you're enjoying spending your time?
Yes, yes, very much.
And Stephen, from your perspective, how have things been over the last few years?
Well, I guess I'll prompt Rachel for things she said before this year. But one of the things you said was when you went on the trial, you felt like you were doing something. At least you could do something. So when you have this diagnosis, it's pretty devastating. And then you assume that there's this decline. And so as far as we can see, we're bucking the trend, which is absolutely amazing. So I think
that's overstating it a bit. I think I'm holding my own. I'm not going to...
I think that's bucking the trend. No, so I mean, Rachel will ask you the same question 3 or 4 times in a conversation, but I mean, that in the scale of things, that's nothing. So we did a load of prompt notes just in case we're dry, but we have a very good quality of life, and that's what you're, I guess, interested in.
Yeah. So you've been part of this trial for many years. Roughly how many visits do you think you've had?
Well, I must be 60 because she takes 5 years times monthly visits, 12. It must be 60, so ish.
And I mean, how's that been? I mean, this is something that we may be looking at on a large scale. How have you found it? What's been good? What's been difficult? What's been easy? And what's been not so easy?
Well, what do you find?
Well, it's never nice being having something stuck in you. But once it's in, it's good. And it's a very nice place to go to Wolfson.
Okay, so the environment matters.
Yes, and all the staff are lovely. They're very, very supportive. And that helps a lot.
And scans and so forth?
Not keen on some scans.
Yeah.
And what I would say is that the Wolfson, because we've been going there for quite a long time, and the MRI machine used to have a little mirror in it, and it doesn't anymore. That makes a big difference.
Okay, so that's something.
Those little things
That matter.
Yeah.
And in terms of side effects, I mean, that's something clearly people are concerned about. And of course, we'll say from the start that everyone's experience is different, and your experience will be different from those. But from your perspective, in terms of side effects after injections and infusions and things?
I mean, I'm not keen on injections, but you know, they're fine.
You tend to like to be calm for the rest of the day, don't you? So if we have an infusion in the morning or the afternoon, sometimes we've gone out in the evening, and then sometimes it's okay, sometimes it's not. You feel really disorientated, or you just feel whacked the next day. But it's not predictable. So actually, sometimes you're absolutely fine. And you did have brain bleeding, I think, in 2017, I think. So, and there was a slight worry more from the clinic than from us, and then we skipped an infusion or two, and then we got back. And so there's not been any more recent history of that we know about.
Sure. Well, I think you'll probably be told
if
there was something to be
told.
And did you have any symptoms at that time?
So what do you mean?
When you had the problems earlier on, did you have any symptoms at that stage?
No, I don't think there's any different for you as an experience. It was merely the fact that the clinic was worried.
Okay. And from Stephen, from your perspective, I mean, Nick's spoken about the huge benefits of having an advocate and someone who can do. What's been made things work or not work, or been difficult or good for you in terms of what we can learn in setting up sort of new services?
I was very struck by what you said at the end of the previous session. It seemed totally obvious, but it is very much a partnership. I mean, it's really good for me. So obviously, it's a double act. And I think you feel supported by me, and the other way around, I feel totally involved in it. And I mean, that's the other thing about the trial as you are, and the monthly infusions. If you're going to somewhere that's completely supportive, and people are very empathetic, as they uniformly are at that place, then you're in touch. You know, you're not remote. So I told you earlier, we had a colleague who had an Alzheimer's diagnosis at the beginning of the pandemic, and they never got to see anybody. It was all done on the screen. And that was a rapid deterioration, and you think our experience is kind of quite wonderful compared to that.
And in terms of, I'm not prompting you, but you're an architect, aren't you?
In terms
of, I am
but I said something before.
You said something before.
I mean, every time we go there, I'm struck by the fact that somebody really, a few people, really cared about the way that place is. There's been a certain amount of money invested in it. It feels like it's really there to work well. Everybody's taken a great deal of care with finishes and junctions and all that. I'm an architect. but I believe, I don't think it's just architects who believe that environment makes a difference. So it's full of light, and it just feels like people were really serious about the mission of that place. So that's why.
That's why I asked you for that.
But also the staff at one.
Oh no, totally. But then that's a platform for the staff.
Yeah.
Things that you don't like.
I don't like the initial when it goes in, because that always hurts.
But there's another thing you really don't like, which is...
Oh, the lumbar puncture, yes, I do that.
That's
really.
not a fan of that.
And they're terrifying, and I think it's, I'm glad I'm not having any of those.
I think you found PET scans worse than MRIs, is that, or did I imagine that?
Yes, MRIs are not so bad.
Is there anything else that you'd like? I mean, we've got an audience here which is really wanting to sort of deliver these sorts of treatments, and we're not the experts, but you guys really are. So are there things that you would like to tell us that we haven't mentioned before that you think it will be beneficial?
Well, I guess in terms of the way into this, I think we were very fortunate. I mean, we've been bloody lucky, if I may say so. First of all, you find out about the trial, which is fantastic, and then you've got to have somebody to talk to about whether you can get on the trial. And then you've got to qualify for the trial, which means you've got to be a bit ill, but not very ill. And then you've got to be sufficiently, and then you've got to need a second person to be a witness. So, and then once you've gone off, and you have to have no conflict with your other medication. So Rachel was on Donanemab for osteoporosis after chemo, and that could have killed it. So we've gone through a lucky few gates. But to underline the point that was made earlier, if we didn't read the newspaper, if we didn't happen to work out how to navigate with that, then it just wouldn't have happened, and we would have been on a different trajectory.
Good. I think on that point we'll stop there, and thank you both very much.
I think if ever there was an incentive to make sure our trials work right, we look after patients right, and we get treatments for them, that was articulated so beautifully. Thank you so much, both of you, for talking with us. 60 visits. And in spite of not liking lumbar punctures, comes every month. So it's amazing. Right, I've got 15 minutes to tell you about the implications of disease-modifying therapies for clinical services in the UK. Not long, so I'll see how I get on. But basically what I want to try and do is raise some questions in your mind so that we can have a good discussion at the end. Currently, our clinical environment is really not a place where treatments can be given in the way that you just heard, in an inviting environment where you feel safe, where you have the experience to be able to give disease-modifying therapies. We have under-resourced memory services. They're fragmented. They don't have sufficient access to investigations. And most of them are community-based and based, understandably, on supportive and palliative care primarily, with emphasis on overt dementia, MCI. Often people are not allowed to follow up. So the very population that we really want to focus on for clinical trials, we aren't seeing regularly in those clinics, at least clinically. We might be seeing them in research studies. The conceptual framework from the public and also from some professionals is dementia is a disease of old age. There aren't any treatments. You're going to get worse. So it's untreatable. And these drugs have given us the incentive and the opportunity, regardless of the size of the effect. The fact that they exist has given us the opportunity to really change that and to reframe what we do with our dementia services. AI UK did a survey in 2021 of local memory service consultants who are primarily old age psychiatrists, asking if they felt ready to give disease-modifying therapies. This was when Aducanumab first came out with its accelerated approval, so that was the first flurry of trying to understand disease readiness. And most people were very clear that they didn't have the necessary skills to do this. They didn't think they could access the necessary resources. They didn't think they could probably do it in a year, but they were really keen to get those skills and to be able to deliver those therapies. And I think that the time that they felt was more realistic was more like 5 years, not 1 year. With the current capacity in this country, just in terms of specialists and in terms of infusion capacity, never mind investigations, which are equally important. This is a graph, and I apologize for the tiny font. Basically, this is the RAND Corporation, did some modeling in the US and then did the same modeling in 2019 in 6 European countries. So you've got France, UK, Sweden, Germany, and I can't remember what the bottom two are. There you go. Red is dementia specialists, and green is infusion capacity. So with our current capacity in the UK, and we're in the top right corner, it would take you 14 months to see somebody for assessment for disease-modifying therapy. Even once you've seen that person, that green tail is a backlog of you waiting to get your disease-modifying therapy because there isn't any room in the infusion suites to give you it. So you would have to wait till 2042 to clear that backlog. And that means 1 million people with MCI would get to dementia while they're sitting on the waiting list. And that's just not acceptable. So we have rate-limiting steps in every part of the pathway. Not enough workforce, not enough delivery, and not enough investigations, as we've heard about already. So we really need to think smart about how we reshape our service. We need to focus on early diagnosis that is path-specific, because these drugs clearly target one pathology. We need to have a much more integrated, joined-up, multidisciplinary approach, not the fragmented approach that we have across the different trusts and the different ways of running memory services at the moment. We need to align our clinical and research. At the minute, quite often, they're very separate. It's not often that places completely embed them, and that's so important to improve both of them. We need to be looking earlier at prevention, and Gill's here who did the Lancet Commission on prevention. We need to be putting that within our clinics or in some form in our brain health clinics and managing prevention as well. So moving from that palliative model to a chronic disease management MS-style model is really where we need to be going. And that's a big step. But the time is right. There are a number of international initiatives at the moment, and you heard earlier about the mission from Hilary in her reflections, and that's a really important statement by the government that they are prepared to put funding into changing what's happening at the moment, which is just as well, because there's almost 1 million people in the UK with dementia, and if you put a finger in the air and get a rough estimate and take it with a pinch of salt, between 300 and 500,000 people in this country might be eligible for disease-modifying therapy if it's MCI or mild Alzheimer's disease, MCI due to Alzheimer's disease. So somehow we need to change our systems so that at some point we can meet that demand. I don't think we're going to be able to do it next week. So what do we need? Changing the culture so that we focus on early diagnosis and are comfortable making an early diagnosis. And a lot of people in local memory services are not comfortable. So 40% would not give a diagnosis of Alzheimer's disease to somebody before they got to the threshold for dementia. And several reasons are given for that. A lot are poor access to biomarkers, difficulty confirming diagnosis, but also worry about the negative impact on participants or patients. And actually, if you ask patients whether they want early diagnosis, 75% say yes, please, even if there isn't a treatment. Whereas people think professionally that that might be a more negative thing. And I think that's a conversation to understand much better in terms of where public perception is and where professional perception is. And in order to make that early and accurate diagnosis, we have to have biomarkers. We know that amyloid positivity is required for these disease-modifying therapies, and studies like the IDEA study have shown us that we're not that good at diagnosing Alzheimer's disease. They did PET studies in people with a clinical diagnosis of MCI or mild Alzheimer's disease. It changed the diagnosis in 35%, and if you looked at those that were amyloid negative on the PET, so did not have Alzheimer's disease, 72% were diagnosed with Alzheimer's disease. So it's critical that we use these in our early diagnosis, and the studies are hopefully pushing us that way. In trials, fine, you've got amyloid PET, tau PET, CSF. We have not got enough PET capacity or not enough money in this country to pay for enough PET to be able to use that to diagnose everybody that is potentially eligible for these treatments. In the short term, CSF is a more reasonable option in terms of being able to upscale it, although clearly it's an invasive test. It's something that needs to be expertly done. You can tell not everybody likes it. Rachel really doesn't like it, though she still has them, comes every month, so she's prepared to put up with it for a purpose. So we need to upskill our local services so that they can actually perform these tests to show amyloid positivity. And for that, we need more labs. We've got one lab in the country that manages to analyse CSF for neurodegenerative markers, so they're not going to cope with an increase in demand that we're talking about. In the future, we'll be looking at plasma biomarkers, and that's not far off, but we are not quite there yet. And you heard about the specificity of P217 earlier. That's very exciting in terms of showing AD, but at the minute, that's a triaging system. We're not quite there yet, but we will be there soon. Unfortunately, biomarker access is very inequitable. So it's great that in local memory services they have better access to MRI than they used to. It used to be CT if you were lucky. But there is very little access to PET or to CSF at the moment. They can't directly perform it, they can refer for it. And there again is a discussion about what you have within your service and what you might have to get from outside. So we have a way to go for biomarkers. What about neuroimaging? You've heard from Frederik about neuroimaging. It is critical for diagnosis. It's critical for safety monitoring. Nick and others have mentioned ARIA, the commonest side effect of these monoclonal antibodies. They're not trivial. You'll see down the bottom in terms of the numbers, the percentages of people in the trials that have got ARIA. But as you may be aware, most of them are asymptomatic, transient, and go away on their own. But rarely they can cause serious side effects, and you need to be able to pick those up. In the real world, that's going to be much harder than in a sterile trial situation. You need adequate MRI access. You need neuroradiologists like Frederik that are going to interpret your scans, and you need clinical expertise on picking up fairly soft, non-specific issues that people have when they are symptomatic. So it's critical that we have that resource. But we don't have enough MRI scanners. We were on a meeting earlier today, we were talking about we do not have enough MRI scanners to be able to scan these people. So what can we do to try and improve access? Well, this is one possible innovation solution for one part of this problem. We need multiple innovations here. So this is B-Rapid and ADMIRA. This is a study that Nick and I and Jeff Parker are involved in, where what we do is you come to clinic, you have your one-stop shop, clinical examination, psychology, MRI, and while you have the MRI, we ask you if you want to stay in for 5 minutes more. And we do some rapid sequences within that extra 5 minutes interleaved with the scan. We also take a blood test for biomarkers. 95% of people say yes to this. 5 minutes more in a scan when it's 30 minutes is really not a big deal for most people. And the point is that on the left is your long, complicated pathway with very expensive PET and CSF and a long scanner that people struggle to tolerate. And on the right is your very brief scan with a blood-based biomarker, and potentially in the future, if you can show that you can get a diagnosis with those that isn't inferior to the complex pathway, potentially we can use that with algorithms from machine learning to semi-automate it. So we need less of Frederik to interpret all of those scans. We'll still need Frederik. He'll just be supervising the machine. Sorry, Frederik. This is an example of the scans that we're getting from that, and this has now been going on for a year. And just quickly to show you, so one of those scans is the rapid one, it takes 5 minutes, and one of those scans is the one that takes a lot 1 minute, and one of those scans is a lot longer. And I'm not going to make you guess, but basically, no. Nick always makes people guess. The one on the right is actually the short one, and it looks better. It certainly doesn't look worse. So we're showing that these scans can work, and we need to roll this sort of thing out to make scans more accessible. Future pathways for AD and trial readiness are very hot topics right now across the globe. This is based on a paper written by an international group of experts that tried to look at a pathway. It's actually a relatively simple pathway compared to mine, which looks like spaghetti junction, but I'm not going to show you that one. So thinking about prevention, brain health centres or wellness or in primary care, we need to be focusing on risk reduction, identifying those risks early on and ensuring people optimise their resilience. Once they get symptoms, they're going to need to be identified appropriately in primary care, and primary care is going to need support to be able to do that. And we're going to need to work much more closely together than we've been doing so far. There'll be an assessment that eventually it might be digital tools in primary care, it might be blood biomarkers in primary care. We're not ready for that yet. So it will need to be a joint assessment with the appropriate people going for secondary assessment in secondary care, having biomarker diagnostics, coming back to the point that these are inequitably accessible, so how are we going to do that? If they're biomarker positive, we'll need to judge eligibility. And there's a huge list of things that might exclude somebody from having treatment. We need to go through that, not least having a Steve to look after you and partner you when you go to have your treatment is something that's really important. But once somebody starts that treatment, they're going to need support. They're going to need monitoring with MRIs, with regular clinical assessments. And we're going to need to decide when to stop. What we've forgotten about is there's a huge number of people that aren't eligible for these treatments because of comorbidities, pathologies, they haven't got a Steve. Those people need an equally important pathway of care that we mustn't forget about. And those that don't have Alzheimer's disease, that have other diseases, also equally importantly need those pathways. The problem we've got is that at the moment the NHS is in crisis. We're talking about this really exciting opportunity. It's absolutely fantastic that we've got this opportunity. It's a brilliant problem to have. the NHS might as well have a note written on the desk saying there is no money left. I mean, it's difficult at the moment. It's challenging, and this is not going to be cheap. And it's going to need a lot of resource. So we're not going to be able to set the whole thing up all in one go, because it will fall over. And some people won't be ready for it, whereas other sites will. So we need to think cleverly about how we can start things in a staged approach, and how we can build and learn and educate those that have had less opportunity to understand the various things I'm talking about, like biomarkers, and develop those blood biomarkers and scale up the cheap, accessible measures. And I think the way to do that is you start looking at the sites that are research active. You start looking at the sites that have been using these drugs in trials involving people like Rachel, that understand them and understand the difficulties of them and understand how to pick up the subtle changes. You start with those sites, and those sites then start to train up other sites and upskill them. And part of that potentially is this Brain Health Centres network, which involves an increasing number of sites that are now embedding research in their clinical, which is already increasing quality of the care within the clinical service and increasing their ability to do things like CSF. I think if we do that, we will be successful with these treatments, we will be safe with these treatments, but it's going to take a number of years. But it also gives us time then to get the blood biomarkers and the other things that we need to make sure that we can do this on a UK scalable manner. So in addition to starting slow and building slow, I also think it's critical that we have training not only for our professionals, but education for the public and for our media. We need the right messaging to go out. People need to have their expectations managed. These drugs are not for everybody. People need to understand what the risks and benefits of these drugs are and how to explain them. We need to make sure that our pathway design is sensible and logical and makes points of all the things that you've heard already today and we'll hear further in this session, and learn from other specialties. We're not the first people to have found a drug that actually might do something, so we need to listen to the people that have been grappling with that for decades. We need novel tools that make life easier and more accessible for everyone, and we need to have those registries I was talking about earlier to make sure we understand what exactly we're doing with these drugs over the much longer term. Money is always difficult, but if we can think smart and take it in a staggered approach, I think we can manage that. We haven't got an option. We've got treatments for the first time, so we have to do this. We just have to work out how to do it. And we have to work together as a network. So I think it's a really good problem to have. I think we need to grasp the nettle, but I think we need to walk before we can run and do this in a careful, considered manner. Thank you very much. So now I think, John, we've got you. John's going to talk to us about what's happened in the States so far and what we can learn from them, which I think is a huge amount in a relatively short period of time. Thank you very much. I have the slides. Where are you? Yeah.
Well, thank you so much for the invitation to speak today. I want to thank the organizers. I especially want to thank Rachel for participating. I think that was really special and important to hear your voice in all of this. So today I'm going to talk about what the US is doing and also how at Mayo Clinic we're approaching this. So I work at Mayo Clinic in Rochester, Minnesota. I'm a behavioral neurologist there. And in one of my roles, it's to prepare us for rolling out Lecanemab. I have no relevant disclosures. I do serve on the Data Safety Monitoring Board for a StrokeNet trial that's unrelated, and I'm a site investigator for the AHEAD study, which uses Lecanemab in a different population. So what's the current state of Lecanemab in the US? So in January, as you heard, it received accelerated approval with the FDA, indicating that it effectively removes amyloid from the brain. But it's still waiting for full approval, which we anticipate the decision to come later this year. But in February of this year, the Veterans Affairs Hospital System published criteria for prescribing to their patients, and they included some interesting provisions, such as they're treating people over 65 and they're requiring a specialist provider, geriatrician, geriatric psychiatrist, or neurologist. But this is a large health system within the US that is starting to move forward with treating folks. And then shortly after the FDA's full decision, we're going to hear from Medicare or CMS determining how much they will pay for this type of therapy. What are the anticipated barriers in the US? Well, it's very similar to the barriers that I've heard about today that are occurring in the UK. So there's a lack of dementia specialist supply. That same RAND analysis anticipated up to an 18-month wait time for a specialist in the US. We need access to neuroradiology, neuropsychology. Our infusion centers currently don't have capacity. We have limited MRI availability. And one of the other issues is we have a large rural population, and as you heard, for this drug, you have to come in every 2 weeks, and there's a lot of safety monitoring, so there's going to be geographic limitations for people. And then, as I heard earlier today, we also traditionally diagnose someone and send them to be managed more locally, and we need more capacity for these follow-up evaluations, as well as access to PET scans and biomarkers. So now I'm going to go through some questions that we asked ourselves once we heard about the Lecanemab news. And the first one is, should we treat with Lecanemab? And so the way we decided this at Mayo Clinic, we have 3 main sites, the Minnesota site, the Florida site, and the Arizona site. We all got together, the specialists, and while we have differing opinions on a variety of things, with this we were all in agreement that via a shared decision-making process that Lecanemab should be offered to a subset of patients. And we came up with a plan to develop a comprehensive clinic for evaluation and longitudinal implementation of Lecanemab, but other emerging therapeutic options for Alzheimer's disease as well. And we formed a project team to put this together, of which I was able to participate in, and we're still working on it. The next question we asked ourselves is, who should be treated with Lecanemab? So we currently feel like it should be restricted to people who are in the clinical trial because that's who we know the safety data from, and not to extend it to people who are earlier in the disease process or later in the disease process, or who have comorbidities that weren't tried for safety. We then wanted to determine how many people are we going to be dealing with that will meet this clinical trial criteria. So we have a population-based study in Olmsted County, Minnesota, where we evaluate people with this detailed biomarker testing, amyloid PET, MRI. We get a lot of their medical history from the records. So this is representative of the Minnesota population. So we follow 2,100 people who had amyloid PET scans. About 870 were amyloid positive. And 237 of those had mild cognitive impairment or mild dementia. But once we put them through the clinical trial exclusion and inclusion criteria, only 19 patients don't have any exclusions. So you can see that this is very exciting. The number of people who qualify ends up being really low. And then we decided to do a more liberal approach and say, we're going to not use the same mild cognitive impairment criteria used in the trials. We're going to use a larger amount of people. You might be able to double that number, but it's still a small number of that total population who qualify. When you look at why they were excluded, it's the most common reasons are imaging abnormalities. So these people have too many microbleeds in their brain, multiple strokes, a large amount of white matter disease, that would have been excluded from the trial. And the second most common reason is cardiovascular comorbidity. So people in their 70s and 80s have a lot of medical issues. And as we heard from our clinical trial participant, you can't be too sick to get into the trial. And then also cancer, as you can imagine, would be a restriction. The next question we asked ourselves is, how do we make administration safer? So we tackled anticoagulation issues, ARIA management, and whether you have this ApoE4 homozygote profile. So if you were in the trial and were on an anticoagulant like warfarin or Eliquis, your rate of hemorrhage was 2.4% compared to a placebo of 0.1%. And if you take that back to our atrial fibrillation trials, which is the most common indication for anticoagulation, the rate of intracranial hemorrhage is 0.24%. So you're looking at a 10-fold difference in risk of hemorrhage. If you're on anticoagulation, you're getting lecanemab. So our viewpoint as we're rolling this out was not to offer it to people on anticoagulation, and the appropriate use criteria that were recently published had a similar viewpoint. We're going to monitor ARIA risk similar to how the FDA recommended. We're not going to treat people who have more microbleeds than were allowed in the trial, but we wanted some additional exclusion criteria to make it safer. One of that was excluding people with anticoagulation, as I mentioned. The second is people who are ApoE4 homozygotes who already have a microbleed, where we don't feel like there's enough safety data on them to initially offer this, but would be anticipating additional data to see if that would become safe. We also know that almost all the side effects from ARIA occur in the first 6 months, and they occur in a dose-dependent manner with your ApoE genotype, so we can leverage that information to monitor our patients safely. So we are going to have a normal risk pathway where people who have no ApoE4 allele would be monitored similar to how the FDA recommends, but a high-risk pathway as well for our ApoE4 homozygotes who we know are at much higher risk of ARIA, where we might get an extra MRI scan. They're going to have more high-touch visits, and we already know that's already a big burden, but we feel like for this to be safely rolled out, we need to carefully monitor this group. We've learned a lot from the publicized cases. Nick mentioned one case of someone who got TPA. There was another case of someone who was on an anticoagulant but got a hemorrhage. And then there was a third case of someone who was an ApoE4 homozygote who was having symptoms and got infusions and had severe reactions. But if we can avoid anticoagulation, educate our stroke colleagues who are giving thrombolysis about this medication and the potential interactions, this would make administration safer, and that's one of the things we're doing. So what's the most appropriate amyloid screening test? This has been covered by a number of people here. CSF versus amyloid PET. Currently, CSF is paid for in the US. Amyloid PET is approved but not paid for. So we're going to also start off with CSF. Amyloid PET does have some advantages with quantification and monitoring, but we're anticipating that it may be covered at some point, but we plan on using CSF. We've thought about plasma, but we also agree it's at the screening stage of the disease. Maybe when combining it with MRI and AI, we can get it better. But this was a publication from our group, and you can see that factors like chronic kidney disease, stroke, myocardial infarction, and BMI all change your plasma value. And if we're going to give this serious medication, we want to be very sure that we're treating the right disease and don't want these comorbidities to be involved. So I'll finish off by talking about the implications for our practice in Rochester. So even within Orange County, we have 160,000 people. So that means there's 9,000 people over 50 with MCI or dementia that may need to be screened. And so there's a large eligible population, but as I showed, only a subset of those would actually be eligible. We've engaged these stakeholders with regular meetings to make sure that they're involved with the planning and implementation of this. I'll highlight with radiology, we are also working on an abbreviated MRI protocol so we can get more people in. With neuropsychology, an abbreviated profile to get more people through. And then our primary care doctors and geriatricians discussing the criteria for patients to be referred. So if someone's not going to be eligible, to make sure that we're not getting those patients referred and they're discussing options and care management pathways with them up front. So this is what it looks like for us. You have your scheduling arm, where people can come to our clinic externally, internally, from other neurologists. Then within a 4 to 5 day period, we're going to get, hopefully, neuropsychology evaluation, neurology evaluation, ApoE testing, blood work, lumbar puncture. They'll meet with our social worker who will discuss cost, which will be very important, and then we'll have a multidisciplinary case conference to discuss each of these cases and can come to a consensus recommendation with everyone's input. And then they'll move to a treatment arm where we can scale because we'll have nurse practitioners involved in the infusions and monitoring, and the neurologist will be carefully involved when and if ARIA occurs. So this is the current plan. So in summary, we're excited that disease-modifying therapies are here. The numbers, the demand is staggering, but if you look at the people actually eligible by the trial, it actually ends up being much less than you would think. And we really think a multidisciplinary approach is the best way to deliver this complex medication safely and effectively. Thank you very much.
Thank you, John. That was such a practical illustration of what we all need to be thinking about. That was really, really useful. I've taken lots of photos. So moving on to Heather, who has long experience personally and also within the specialty of using disease-modifying therapies within MS. And thank you so much for giving us some highlights of the lessons you've learned.
Pleasure. Thank you so much for inviting me along. Actually, it's a really exciting occasion, isn't it? And I've been given 10 minutes to tell you all about my experience, my similar experience with MS. And I think I'm on. Oh no.
You can talk and we'll sort it for you.
Okay, lovely. So there are a lot of similarities. The situation we're all finding ourselves in here and the situation we found ourselves in the MS sphere 30 years ago now, around about. But there were a lot of differences as well, and I think it's really interesting to... hopefully you'll get some ideas from our experience. Right, let me see if I can work this. So first thing to say, 2.8 million people worldwide with MS. So the numbers are very different. And there's the equator. The dark red is the areas of particularly high prevalence, and UK. We've got 131,000 people. This is from 2020, these data. 131,000 people living with MS in the UK. That's 1 in every 500 people. And 130 patients are diagnosed with MS every week. So that gives you an idea of the scale. What are the impacts? Well, other than the obvious impacts of accumulating physical disability, cognitive disability and so on, there's a huge societal impact for MS. The patients are young, women of childbearing age, people who are working, people who are wanting to start families and so on. So, and if you look at some of the employment data, it's staggering, has a staggering societal impact. By the time someone's been diagnosed with MS for 10 years or reached EDSS 3, and for those of you who aren't familiar with the EDSS scales, EDSS 0 is unlimited mobility and no neurological symptoms at all. EDS 8 or 9 has been very, very disabled, bedbound from MS. EDSS 3 is no walking impairment, but a number of symptoms and a number of signs. Okay, so even when young people with MS, no walking impairment, 50% of them are unemployed. So it has a huge, huge impact, and that's European wide. Here, 150 years from the first descriptions of MS in 1860 until we developed the first disease-modifying therapy in 1993. Okay, well, rather we offered the first disease-modifying therapy. So, a long time. And here, so this is, I was working it out earlier, the 30-year interval here from the first disease-modifying therapy to the current day, pretty much. So this has been a long process over a long period of time. Now, this is where I qualified. And this is where I started seeing patients as a consultant neurologist, started seeing MS patients. So as a medical student, it was quite easy learning about MS disease-modifying therapies. And I've had the fortune, really, of not really having to learn too much before I qualified as a consultant either. And then once I've been a consultant, I've been learning all these new drugs have emerged as I've been a consultant. So I've been drip-fed all this information. And so it's been a really interesting and very exciting time, and I'm really glad that we're in that situation with dementia as well. Now, what did I want to point out here? So beta interferon, glutarium acetate, these are self-injections, about 3 times a week. Quite a lot of side effects, but very, very good long-term safety, okay? This is all we could offer people, and it was, I'm going to call these low-efficacy therapies, okay? We're having the same problem. What is a low-efficacy therapy? We thought it was a low-efficacy therapy. We didn't have, we had small amounts of clinical advantage, but we didn't have long-term data. Now we have the long-term data. I'm looking back going, yes, these treatments are actually pretty effective. The earlier they're given, the more effective they are. But that's the wisdom of, very much the wisdom of hindsight. And the wisdom and the advantage of really careful monitoring of that patient group as part of the risk sharing scheme, which was incorporated early on because there wasn't much data. And the risk sharing scheme was really helpful for us. It was basically part pharma funded, part government funded. It allowed us to provide some of the resources to give these treatments, at the same time monitoring patients and seeing whether there is going to be some long-term benefit. Anyway, natalizumab was the first really of the high efficacy therapies. Natalizumab is a much bigger deal in terms of administration. Beta interferon, glutarium acetate, you give them, you show the patient how to self-inject, you send them home. You do a periodic blood monitoring, and you have a drug delivery, home delivery service to give the therapies, and that's it. So it's fairly resource low, except for the cost of the drug itself. Okay. Natalizumab, something completely different. Infusion every month. The drug's expensive. The cost of the infusions and so on, and the resources required for that, much more bigger deal. I'm going to talk a little bit about the main side effect of PML that has had big implications for how carefully we have to monitor certain people in that natalizumab group. Anyway, that was a great development, natalizumab, and another great development was alemtuzumab, which was the first of the induction therapies. So we're talking here about maintenance therapies, aren't we? Saying, look, this is a patient. Our population of patients who are young, if you're going to give someone a maintenance therapy, you're going to be treating them for possibly 70 years or something. In people, in the group that we're talking about at the moment with you guys is going to be less, shall we say, to put it tactfully, less long-term treatment. So we had a big problem of, okay, we've got lifelong treatment we've got to manage here. So alemtuzumab, an induction therapy, you give a course of therapy and then that's it, finish, and you just need to monitor afterwards. So induction therapy is very, very appealing for us for all sorts of different reasons. Alemtuzumab, as you know, was given by infusion every day for 5 days, nothing for a year, another 3 days of infusion, and then finished. But patients have to have monthly blood tests for 5 years. 30% risk of thyroid dysfunction as a side effect, for example. A small risk of thrombocytopenia and renal disease as well. So not perfect, but a real great development. And the next one I wanted to just highlight here, and there were plenty, as you can see, but cladribine, Mavenclad is an oral induction therapy. So tablet every day, or 1 to 2 tablets every day for 5 days, nothing for a month, another 5 days, nothing for a year, 5 days of tablets, repeated again. So, and then finished again. So even more convenient, you don't have to have the infusion cost, you don't have to have the concerns of longer term management. So we've got all these options now, and they just crept up on us over what seems like no time at all. And then now, of course, we have the CD20 therapy. So ocrelizumab is given by a drip every 6 months. And now I'm putting them here. The colour coding is also they're high efficacy therapies. They are much, much more effective than the original beta interferons and glutarium acetates, which we now call kind of platform therapies. And then the last one I wanted to mention was, so ofatumumab is a CD20 therapy, but again, it's given to patients give it themselves once a month at home as a subcutaneous injection. So again, far fewer of those implications for some of the ways that these drugs are given. I'm not going to frighten you too much with these are our currently available disease-modifying therapies for MS. And I've just kind of colour-coded them a little bit in terms of efficacy, really. Efficacy, here the low efficacy, medium efficacy tablets, maintenance tablets, people take them every day. I won't go into details because I could talk forever about this, and I won't do that. I mentioned cladribine induction tablet course already. I mentioned natalizumab. monthly infusions, and these are the other. And I've got stem cells at the end there, which I'm not going to dwell on, but of course that's another treatment that we're beginning to look much more seriously at, which I could tell you all about if I had more than a couple of minutes to do so. I wanted to bring up PML, because this, I think, is similar to the sort of experience you're beginning to say, okay, well, look, we've got some potentially serious side effects with our drugs. What are we going to do about that? So this sort of happened with us in a similar way. So patients who were started taking the Tysabri, a few patients developed PML once we'd had a decent number of patients on the treatment for a decent amount of time. And of course, this was dreadful. We didn't know what we were dealing with at the time. I'm sure Frederik probably remembers it well. And when it became clear this is what was going on, we then managed to adapt in quite a successful way to identifying patients who you want to put on Tysabri, but who are high risk of, who are either at high risk of developing PML or who are at very low risk of developing PML. We wanted to identify those patients to decide, do we give them Tysabri, which was at the time really the best, most effective drug, or do we not? And we managed to, over time, that developed an assay looking at JC virus serology. And then subsequently, and more recently, actually fairly recently, we've looked at titers of JCV antibodies, and we've been able to stratify the risk of a single individual patient depending on what their JCV serology titer is, how long they've been on the treatment, and so on. So, and the JCV PML risk, if you're negative for the antibody, your risk is 1 in 10,000, which is similar to I think my risk. And that's, I should say, I'm not taking Tysabri. Whereas if you've been on Tysabri for 2 or 3 years and you've got a high titer of the antibody, your risk is 1 in 164, which I think we all agree is utterly unacceptable. If you identify patients who are at high risk, then they have to have really intensive monitoring, and that includes 3 monthly MRIs. Can you hear me all right? Oh my God, I'm nowhere near. I've got 1 minute. I could go on for ages. I'm going to move on. Okay, so these are the questions I got asked. I'm really going to... Can I have a couple more minutes? These are the questions in 10 minutes. So let me just say quickly, I don't know if you're familiar with the Getting It Right First Time initiative, which is a national initiative, which is really great, and there's a website that's got all sorts of very interesting data. These are the neurological sites in England. What happened when MS services were first introduced was we identified the different neuroscience, the neurosciences centres, the neurology centres, the DGHs with neurology input, etc., and these are all colour-coded here. We set up, so we identified them as, I say we, as specialist MS prescribing centres. We had to identify prescribing neurologists, clinical nurse specialists needed to be brought on board, really key point. We needed to, as the administrative burden became more and more clear, we needed to get MS coordinators and administrators, pharmacists were invaluable, and then we set up the multidisciplinary team meetings where we look at eligibility. So it's very similar to what you're talking about, eligibility of different patients for different drugs. Some of that is mandated by NHS England with this algorithm. There are international and there are national guidelines as well, but we obviously follow NICE guidelines for eligibility, and I won't go into detail. Every MDT needs two consultant neurologists, a specialist nurse at the very least. I put neuroradiologists in brackets. We need neuroradiology, but we have a separate meeting for that. Look at the variation in prescribing practices. Pink is high dose, high efficacy therapies. Blue is low efficacy therapies. That's across the UK. Very, very huge differences in prescribing practices and costs. And I should say £0.3 billion a year is the cost of the DMTs for MS in England. This is our local experience. We've got 5,000 patients with MS, 2,500 exactly on DMTs. And this is kind of what we currently, this is the latest data, this is what we currently prescribe. This is our MS team. We've got 4.5 neurologists, whole-time equivalent. We've got 10 specialist nurses and so on. I'm going to whiz through. Royal Free have, I've got some data there because I was at Royal Free first. Just a couple of other infrastructure considerations. SPRs, succession planning is really important, training is really important. So having SPRs on board is so important, because especially when you're thinking about expansion of your numbers, which you are with your service, you really are going to need to do some training. You've already talked about provision of outpatients. A lot easier now. We have some remote assessments, a bit easier for us in some cases. Day case capacity for infusion is really important. Sorry, I'm whizzing through. We also have a huge imaging burden. I'm sure Frederik will agree. But again, we have very much managed to shorten the length of each MRI, and we've got very specific, which Frederik can tell you because I'm not sure I fully understand. We have a very short scan, which takes about 10 minutes per patient now, which has gone down from about 30 minutes per patient before. So we've made some. We're very, very dependent on neuroradiology support, but we could talk about that at some other time. IT support, having a database of the patients is really important. It's keeping an eye on the safety side of things, and actually in terms of incorporating research and clinical outcomes and so on, is really important. And with the sort of service we have, and I think it would apply with dementia services as well, if you've got an active treatment and you've got some kind of serious side effects potentially that could emerge acutely, you absolutely need urgent access, a very sound urgent access ability. So you need to have a helpline or something, where single point of contact where patients can contact if there is anything serious that happens, and we've certainly experienced that with MS. And then we're really keen to, we're very lucky here, we do incorporate audit and research into our service. This is the last slide. So a couple of other considerations I just really want to quickly say, penultimate slide, sorry. We had some difficulties, what's the recommended caseload per nurse and per consultant? The voluntary organisations were very helpful from that. What are the estimated and projected populations? Again, we had a lot of help from pharma as well as some of the voluntary organisations. The costs versus the benefits. The benefits are, I mean, as a clinical service, these kind of conversations have probably happened elsewhere, but I think it's sometimes it's difficult to quantify the benefits of a successful treatment, and societal benefits and economic benefits, obviously spread across the. Well, this one I just quickly wanted to show you, because actually this is when I qualified here. This is when someone gets to the age of 65, the risk of them needing a stick for mobilising at the age of 65. Pre-DMTs, 80% of people needed a stick. With the advent of high efficacy DMTs, 20%, 30% needed it. So there's a huge improvement, but it's only been proven after many years looking back and looking at big numbers of data. This is my last slide. So this really summarise what I wanted to say. I could say so much more and wish I could, but I accept I can't. So yeah, maybe we can have a discussion about that later. Okay, thank you.
That's called trying to fit 30 years of multiple drugs into 10 minutes. Well done. Could everybody come up to the round table, please? Frederik, are you coming up? Nick, you're chairing that.
Yes, I am. So, chairs. You were thought of. Well, I'm going to suggest everybody feel free to stand up for all those deep vein thromboses that are building up. So you don't have to go on your anticoagulants, so you won't be stopped from taking your immunotherapy. So feel free to stretch just for a moment, take a deep breath after all that. Well, I think Heather's given us a fantastic glimpse of amazing to see that, particularly the level of disability. That's a transformation, if we could see that in our area. So this next phase, we'll try and, again, encourage people from the audience to ask questions. We've had some great provocative and informative talks so far. And I'm really going to try to look at what are the implications for clinical services. And as the last roundtable showed, we will stray widely off topic wherever we're allowed to. So do feel free to that. So Implications for clinical services then. I mean, it's just, I have to say, hearing, great to hear what MS has done. It's fantastic to think about what the US is ahead of us in terms of preparing. It is a little bit frightening to think, how are we going to do this? How are we going to, even with the vastly reduced numbers? And of course, there is this who's eligible, but also who wants it. So in the spirit of avoiding your DVTs and also audience participation, a major issue here is who will be eligible, how will we diagnose them, how will we deliver these therapies. So I'm going to start with an audience participation question. In the UK currently, we have estimates of how many people we think have dementia. And we also have government targets to diagnose all those people in a timely fashion. So you get one vote. In the UK, the most up-to-date figures, is it 80%, 60%, 40% of people in the UK at the moment get a diagnosis of dementia? You only get to vote once. So you've had a moment to think about it. Those who think it's 80%.
Yeah.
Sorry, there's always one. There's always one. Come on. So we have estimates of how many people in the country or in the nation have. And dementia diagnosis has been a target for the government ever since Cameron's dementia challenge. And so that's coded. So a coded dementia diagnosis has been recorded at somewhere in the health system. So how many people get a diagnosis? 80%? Any takers? You've got to vote. There's no abstaining. 60%. 40%. 20%. Okay. The right answer, according to the government, is 62%. Now that doesn't sound too bad. It actually fell since before the pandemic. If you look at what that means, that includes dementia otherwise unspecified. It includes cerebral atrophy, query dementia. It also includes people with Alzheimer's disease. And none of these therapies will be given to people with dementia otherwise unspecified. That's for sure. Okay, so with that little warm-up, we're going to encourage people to call for a microphone for questions, but I'm going to start with a question, which is, very, very briefly, to each panel member, would you go on an anti-amyloid therapy, and if so, when and in what circumstances? Kath. Sorry. That's what comes of being in the office next door to mine.
So if I was affected right now, mildly affected right now, and Lecanemab was available, I would go on.
Okay. Heather, I think you might be...
I think it's an unfair question.
It is unfair. You can pass on that. I think that's absolutely right. We'll have other tricky questions for you. But there is a mic there, John. I think it was.
I mean, it's not complete by chance that I've brought Rachel to talk to us today, because I think actually Rachel and the Rachel situation was probably about the sweet spot, as we've said, where there's definite some problems. They're clear and they're interrupting with daily living, but they're not too severe. When Rachel can make a decision for herself about what to do, lives nearby, and understands the risk-benefit ratio and doesn't have any problems that would mitigate against having treatment. I think in the fullness of time, probably even slightly earlier. I am a believer that the earlier that we go with the amyloid immunotherapy is probably the better and probably the safer. But of course, until that's been trialled, we don't know.
John?
Yeah, so I have a similar answer. If I had mild cognitive impairment or mild Alzheimer's and was amyloid positive, I would want to be offered and would take it.
Frederik?
Yeah, sooner rather than later. With a great prospect as a radiologist to have frequent MRI scans.
We're your friends.
Great, okay. So in some ways I think that sets us up, because what we see on the one hand, we've got our panel who want therapies at the right time, and then we've got a very low level of diagnosis rate. So there are many elements we need to address in order to deliver DMTs, and Kath, I think you showed really nicely what we need, what the issues are, and I thought Heather showed nicely where we might get to. If I ask you to address just one bottleneck that would allow us to get started, I'll open it up to anybody, what would be the one thing that you think we would need to do first, if you like, or the critical path which stops us? Anybody want to take a start at that?
I would like to take a start, even though I'm not in
Yeah, you can get away with anything, Hannah.
Thank you, yeah, I can. No, I think from what I'm hearing about the session just today, I think that the initial diagnosis of MCI, it seems like you've got these huge numbers of patients that you need to screen, and you need to. I was staggered by what you had to say, John, about how many patients there were who were prospective candidates for the treatment. So diagnosing and then.
So the blocked diagnosis being the thing. Kath?
I'm just thinking about what Stephen was saying earlier and how they navigated their pathway because they were particularly capable of doing so, and it's very, very hard to do that unless you're fortunate enough to have those skills. I think there are many blocks, clearly, but thinking about the data that you showed in terms of the small numbers you've got, we've done a smaller study at Queen Square, and actually it is very small numbers in the same way as you've got. So from that perspective, the thing I would want to do is make access equitable and quick and easy. That would be the hardest bottleneck. And then you've got to do the screening, fine, but at least then people are in the system.
Right. I'm happy to. I've got other questions, but I've got a question from the floor. Several. I'm going to start. I don't know, these slackers on the microphones. I'm so sorry there. Do say who you are and then your question, please.
Hi, Susan Mitchell from Alzheimer's Research UK. It was a really interesting session, but I particularly wanted to ask Heather, you talked about all the kind of great things that have happened in the MS field. What was the bit that was really hard? You know, what is the one lesson you might give us for the field? You know, learn from our mistake or our difficulty. Is there one thing you could articulate on that?
Goodness. That's a good question.
Heather, use the mic just because there are thousands of people online currently.
I think probably, as Kath mentioned, I think trying to make things as efficient and simple as possible. I think I'm very struck by the difference in this scenario than the one when disease modifying therapy first became available to MS, and we had this kind of what we thought was a partially effective treatment in the same way that you could argue your drugs are, and we were in this. It was patchy, disorganised. We didn't have any of these forums really to say, okay, what are we going to do and how are we going to plan our services? So I think this in itself is a really
This is probably the most critical step, isn't it? This meeting.
Yeah, thinking about things in advance before that, because we just went along and we said, oh, that went wrong, oh dear, this is bad, and we haven't got any, services, etc.
John.
Could I ask, Heather? I remember also when these first came in, and I think I was working for you at the time. And one of the real positives and things that surprised me at the time was the risk-sharing scheme when it was pharma and the government coming together. I don't know how important that seemed to me at the time to be very important.
Totally was, and I have to say that I think the pharmaceutical industry, fostering very good working relationships with the pharmaceutical industry is key, really key. Working together in that way, I've learned that over the years. As John says, initially they were involved in funding all the initial allowing provision of the B24, and we had a slightly different situation where we had very restricted eligibility criteria for B24, very restricted, but we also had a lot of patients not wanting to have the treatment. It's a new treatment, nobody knew anything about it, nobody fancied it. poor Rachel having the lumbar punctures every month, I can't even imagine. But you know, people were the treatment was self-injection. It was 3 times a week or daily in some cases, and so it wasn't something that a lot of people were very keen to patients were very keen to have. And also, I do think the what's just been said about educating patients and in the community so that they can
very powerful.
Recognise, actually ask for help, or find out where to ask for help.
So, I'm just going to get John, just brief, what, in that same vein, what's your feeling about what will be the, not that, hope you won't be looking back and saying what was the mistake, but what do you worry is the biggest block at the moment? You've thought about it a lot with colleagues. What do you think?
I think I agree with access to an early diagnosis, but that ability to get I think biomarker confirmation is really essential here, and it's not done very widely at this point. And I think confirming amyloid presence is something we learned from the trials that was so important to showing the efficacy. So I think that's another one.
Great. I might come back to that. I've got several people indicating. So I'm going to rely upon the microphone holders to prioritise people, so in the right order, because I won't keep track of that. He's got the mic, so he probably gets the floor.
John Laura, UCL. One of the things that I think comes out as one of the books is about the number of specialists that we have, and I thought, Heather, it was interesting, your last slide, obviously many years into this, thinking about the number of consultants needed per caseload. And, it's fine for us to say we need more specialists, but it'd be nice if we could have a sense of what that number was that we needed when we think about who we need. I just want to know what it was on your last slide, what work's been done on that already, and then perhaps just over to John, whether the US have thought about that as well.
So the original, all those data originally came from how many MS nurses we would need, because when we first started providing treatments, it was all consultant-led, and we didn't have any specialist nurses really to speak of. And so it was all about, the first step we made was we need some nurses on board to deliver the...
Which fits with John's point about the multidisciplinary team being
yeah, and so then the question arose, how many nurses do you need? And actually, if you look at, and so this is where a lot of our voluntary organisations have been really helpful. So MS Society and MS Trust, in doing a lot of work, trying to find out what the optimal workload is for a nurse. And the current number is 315 patients to one specialist nurse. I hear you asked about consultant neurologists. That's somewhat different. They've done we've had a few kind of questionnaires that have been sent out to consultant neurologists in MS, basically asking us, what do you think your optimum caseload is? That's the work that's been done. So it's been subjective, and the number, I think about the average number is around about 600 per consultant. That was a fairly small questionnaire. I'm not even sure it's been published.
So if we scale it up to even just 100,000 people being eligible. Question over there.
Hi, I'm Duncan. I'm a geriatrician from Hillingdon, outskirts of London. I said a question about time frame. Do you have a realistic idea of when this drug might be available to patients in the UK? And I suppose my question is, I'm keeping a spreadsheet of patients who I think might be eligible. Am I wasting my time? Are they all really too advanced?
Who wants to guess that? Kath?
Yeah. Next writer is a guess. So if we think things are happening in parallel in terms of approvals, which is what we think is happening, but we're not certain, then the guesstimate is sort of 18 months, but that's a guesstimate depending on advocacy from charities, depending on patient power, depending on whether NICE decide to play hardball from the point of view of cost, depending on what the cost is at start and whether there is a cost-sharing scheme. There are so many variables. We don't even know whether NICE are actually looking at it at the moment. We think they might be, but we don't know. Unless Alison knows something, she might. Susan, sorry.
Does anybody in the audience know better? Ah, excellent.
Hi, I'm Shahid. I'm the medical director at ASI, and on the NICE website, they've actually got information about the scheduling for Lecanemab, where they're expecting a submission probably around sort of October time. So you've got obviously marketing authorization is one part, and then the NICE submission, which generally takes about a year.
Yeah. So it is about 18 months, so guesstimate.
Good guess.
Thank you. I need to look at the website more frequently.
So since you've got the mic, we're going to give you and you've really provided, what's the question you'd like to ask the panel?
That's very kind, thank you. I suppose, from various presentations around priorities and what to focus on, if you were to have a meeting with NHS England, what would you ask them to be doing over the next year or so? Because I guess the reluctance for anything to happen is because it's still hypothetical. So what would you want them to do now?
That's a great question. I'm going to get everybody to answer that very briefly. I'm going to start with you, Frederik. What would you want them to be doing right now?
I wouldn't know. NHS England. I'm not well that into this.
What would you want them to be doing in terms of imaging?
There's a lot of preparation that can be done. As alluded to, you need to up your game in terms of confirming diagnosis, having probably a lot of conversations with people who are not eligible. What do you do with all those? And then start preparing yourself for, I guess currently you see people once, and if you have a diagnosis, you basically say farewell, unless something significantly changes. And that was the game changer in MS, I think. In the past you established a diagnosis, and then people came in occasionally for a steroid course, and that's it. And now they go on maintenance therapy with, regular visits. So imagine, it's 130 new cases a week, who all come back in a year's time. And this
it's cumulative.
Huh?
Yeah.
And that's going to be, and for that reason, I think it's good that it's going to be introduced slowly. Not too many people will qualify, so you can get used to that. And then once the indications widen or other drugs become available, then it's going to really explode.
Does anybody else on the panel want to have an attempt at that, Kath?
It's a really good question. I mean, we spoke to NHS England for about 2 years from 2021 and had a working group that put together a paper that suggested that we needed to do exactly what we're talking about this afternoon. And it got some traction for a while, and then we started having revolving doors in the government and everything got parked. So what I'd like them to do is restart that conversation and be consistent with it and put time and resource into it, which they were reluctant to do, and there are people in the room that are very aware of what's going on right now. I think that conversation is restarting from what I hear right now, which is terrific, but they need to commit to it being a priority, like they have with the Dementia Mission. And they need to, I think, accept that they have to have oversight as a central body of a drug and process like this, and not just delegate it to the ICBs, which is where it'll get lost in the mud.
John?
Yeah. So I'm not going to talk about the prioritisation, but I think a point that we really need to stress is, I think what we've certainly learned from stroke and learned from cancer, right, and I suspect from MS as well, is that the benefits of getting a coordinated service with good diagnosis, with equity across the board, is likely to improve outcomes for patients whether or not we have effective treatments. So when thrombolysis for stroke came in, the outcomes were hugely better than the predicted benefits from the thrombolysis, simply because there were pathways that got people seen and assessed and diagnosed, and their chest infections treated and their deep vein thrombosis sorted out as well. So there's huge benefits, and I think all of the clinicians in this room would agree that we are not, as a country, offering the best possible service to the patients who have dementia. Nick's diagnosis rates is just one aspect, but in terms of the distribution of care, and we've got a huge amount that we can offer and improve things just by looking after people better, and that's something that I think we can stress as well as as a benefit of getting these pathways sorted out.
A little bit worried when you turned to me and said Nick's diagnosis. I didn't know where you were going with that.
Well, it can only get better.
Natalie.
Thank you, Natalie Ryan, UCL. That was brilliant. Thank you, everyone. I wanted to ask about ApoE genotyping, because clearly that's coming up as one of the major risk factors for complications, and we don't currently do genetic testing for ApoE. John, you shared with me this morning the amazing system that you've developed where people will have a blood test on the Monday and then receive their ApoE genotype on the Friday, which is somewhat different genetic testing timelines here. So I guess my questions are, should we be thinking about testing for ApoE in genotype in patients with Alzheimer's disease? When should we be bringing that in? And then I guess drawing on John's question, is there an opportunity in terms of the work that we could be doing in the meantime for developing links between community mental health services, neurology centres and genetics to work out how actually that would work, because we need to move from people mainly not donating DNA samples at all to a place where certain people would be the ApoE genotyped, and actually that could also obviously have benefits for raising awareness and access to genetic testing for familial cases as well.
Do you want to start, John? I mean, I guess there's the question about the role of ApoE just first.
Yeah, so traditionally we have not done ApoE testing in our patients, and but once we saw the data and how predictive it was of side effects, it really has clinical implications for them if they're going to undergo therapy, especially for those ApoE4 homozygotes. And you saw the VA, for example, may not treat ApoE4 homozygotes because of their high complication rate. And so a patient requires that information to make an informed shared decision-making process. So, I do think in a symptomatic person who's considering this therapy, we should offer it. We're going to really push for it to be done, and then as you saw, they may need to be monitored in a different way. And so what we ended up doing once we were sorting this out is we met with our genetics lab and said, we traditionally haven't ordered this test, but it's going to clinically impact decisions. Can you meet us? And they were enthusiastic to provide that service once we met with them. And so it's something that went from not happening to now, as you mentioned, we're going to try and get it done within a week. And so, and we thought we might get pushback, but they were really excited to help patients, so it worked out.
We've got lots and lots of people who want to ask questions, so I'm going to ask everybody to be brief in their questions and their answers. John.
I think that there's two issues here, which is the rapidity of ApoE testing and whether it's relevant for clinical trials. The answer to that is clearly yes. I do think we also will need to take a step back and just think about some of the ethical implications. These have been written about by Rob Howard and some of our colleagues here at UCL, because of course if you are an ApoE4 homozygote, then your children are ApoE4 positive by definition. And so I think we will need to think about that, whether also there's ways that people, whether in terms of disclosure and so forth, so, Rachel won't know her ApoE4 status but was in a clinical trial, whether there are ways around doing that, the ethics behind that, that's something that I think we do need to start teasing out and working out now while we don't have.
Just a quick point of clarification. You said offer ApoE4, require it?
So, yeah, that's a good question. So from our standpoint, at this point we're thinking of requiring it. It's still in a discussion.
It sounds a bit of thinking of requiring it rather than requiring it.
Yeah.
That's a bit like offering it.
Some things we have like a full consensus of opinion on, but my personal view would be to require it because of the safety issues.
Good. Okay. Question there? Ross.
Yeah, thanks. Ross Patterson, UCL. So one of the most daunting things from my perspective is the caseload of managing people longer term, because we've currently got this culture, particularly in memory clinics, of making a diagnosis, then discharging people at 6 months. So we're going to end up having people in our caseload for potentially 10 years or more, which will be exciting in that we'll be able to look after people really well. But I think there'll be an opportunity to do more than just deliver disease-modifying therapy. We'll also be able to do lots of other things in terms of cognitive support and rehab and occupational rehab. And I guess my point is we need to make sure that we ask for enough. And we've potentially got 3 drugs in the market. So there's potentially buyer's market in terms of negotiating with drug companies in terms of what we ask for to support our service.
Great. So I've got John.
Just going back to the ApoE, I 2 questions, very brief ones. One, does it change your management in how often you are going to monitor people? And secondly, I suppose the parallel I would see is with natalizumab. When I was a registrar, we didn't really have a clue what the risk was. But I suppose the one thing, your amazing picture, that's based on real data, which is, this is your risk. This is what's going to be your risk of lead. I suppose so I see the help of ApoE will be when we have a lot of real data to people, really give people the risk of what their safety data is.
Okay, I'm going to get John very, very brief.
So, yes, we're going to do more MRIs if they're ApoE4 homozygotes to screen.
There was a question where? Oh, great.
Can I just go back to what Ross was saying? I think that's a very good point. And so certainly in MS services, you know, one of the benefits of having that infrastructure and having that big team and having all the nurses and the good links with the rehab services and so on is that your treatment of patients who aren't eligible for disease-modifying therapy is potentially transformed. And I also kind of alluded to that importance of having a good setup for urgent access. So yeah, that's a good
Gina, brief question, well, quick answers, because there's a few more that have come in on my iPad here.
I just wanted to ask about what the long-term implications might be for those subjects or patients who are not eligible for the drug, who don't meet criteria, and also the fact that this is not disease halting. Patients will still progress. So what happens when they go into moderate or severe disease and they have neuropsychiatric symptomatology? Where will the diversion of resources for what is going to be a very expensive and labor-intensive disease-modifying approach, how will that impact on the other patients and on symptomatic modification of non-cognitive?
Okay, so I'm going to just take the last bit of that, which is around stopping. When do you stop? John, do you want to start and then?
Yeah. Well, the open label extension data will be really informative regarding stopping, but in the upfront discussion with the patient and caregiver, we need to discuss that progression will continue and come up with a plan and discussion of when it makes sense to stop. So whether that's going to be 18 months, 24 months, still to be determined.
I've got a question for Frederik. How are we going to deliver imaging? That's the first one. And the second, do you think we're overcautious about microbleeds?
Well, to start with the first one, so I'm glad to hear it will be sort of staggered approach, not too fast.
The reporting power might increase.
You will need, more scan slots. Can be shorter scan slots. You don't need a full diagnostic scan, even short of an abbreviated protocol. A rapid protocol. For MS, as Heather said, we have we just do a flare and a diffusion and perhaps a T2. That's fine, but it will eat some resources. The reporting will take some time. You need to educate people on how to read these scans, what to look out for. And the other thing was about microbleeds. How cautious should we be? Well, that's I think a shifting target. I think the earlier studies were very cautious, and the number has been moving up. But I think only time will tell whether it's safe to loosen up and disregard also incident microbleeds.
Sadly, we're just about out of time, so I'm just going to ask whether the panel have any last comments they'd like to make of any particular, the implications for, back to where we're meant to be, which is talking about implications for clinical service. Anybody, last comments from anybody about what either their concerns or what they would like to see or what the implications will be? Kath?
I think what's been really useful about this discussion is learning what people have learnt before and not making the same mistakes. And having the communication across different specialties, specialisms, and also different countries. You can learn a huge amount. Just because we have different practice to the US doesn't mean we can't learn a great amount from what you just said. So that, to me, is what we need to continue to do.
Yeah. Heather, do you want any final thought?
Yeah, I think it's really, really interesting to see the position you're in now, and it has given me pause of thought, quite apart from having to do the talk. It's what kind of made me really think about it all. And I can't overstress the importance of collaboration on the ground. I hear absolutely what Kath was saying about, we are dependent on NHS England and people telling us, oh, you can give these drugs and you can do that. But actually you can begin to look what your service should be like, and you can begin to work on that, because you're always going to want better services for dementia, quite apart from the fact that you've got a drug that's going to come, and in 10 years' time we'll have you there. So I think
we need to get on with it.
Yeah, I would just say to get on with it without waiting for approval.
Yeah.
Can I say that?
John?
Yeah. And I just think that within my working lifetime, we've seen the outcomes from stroke and from MS completely revolutionised. And I remember for both of those with a massive amount of nihilism.
Yeah.
I remember being a houseman and just begging to get a CT scan for somebody who had a stroke in the middle of the night, and no one was interested.
So what?
And now people are being fast-tracked into hospitals and having scanned and having interventions immediately. We have a healthy scepticism and cynicism in this country, which I like, but that can tip over into nihilism. And I think that we need to grasp this. We need to be very clear, ring-fence, do this slowly and cautiously in the right people. But I think if we could think in 20 years' time and look back about and have those milestones like Heather showed on the MS, that's what we all need. As by that stage, we'll all need those treatments as well.
And there'll be a lot more names to learn. John?
Yeah, piggybacking on those comments, I think the infrastructure we're talking about building is for not just Lecanemab, but for those other drugs that are coming along the way, and to be ready to deploy those in addition to, or maybe they'll supersede.
Frederik?
Yeah, looking back at the MS experience, we started with partially effective drugs, but it's really impressive if you see, 10, 20 years later, how well some of these people are doing. So it's really important to get long-term follow-up data and registries and see what the outcomes are, because you might be surprised and you learn a lot along the way.
Well, since I just saw Rachel nodding along to that, I think that's a good point for us to stop. I'm going to now I'd like you all to put your hands together and thank the panel. And I'm now going to hand over to my colleague Seb Crutch, who will be chairing this next session. We need to gather our round table. Do feel free to stretch your legs momentarily. Oh, there's Kate. Yes, Kate, I'll go and say hello to her. Will you offer her to say anything, or see what she is? I'll ask her what she would like.
Nick, have you got the microphone? Okay, everybody, thank you so much for staying on for this final session of what I hope you agree has been a fantastic symposium. My name's Seb Crutch, I'm one of the psychologists here at UCL. And in these last few minutes, the idea is to broaden out our discussion a little bit to think about some of the wider implications of these disease-modifying treatments. We might be thinking about individual and personal, family, organisational and wide social implications of these treatments, and also what the nature of the kind of hope that we all have. Hope is a word that's come up many times this afternoon already, and what our realistic hopes are, not just for the direct benefits of these drugs, but for their wider implications. Just while our panel settle themselves, I'll introduce them in a moment. I thought it would just be worth sharing a couple of quotes from Kath and Emma Harding's excellent paper thinking about the role of hope in clinical trials, which I think are easily extendable to the situation of treatments, and a couple of comments from trials participants about what extra they get from these trials beside the drug. So one person said, "I joined the trial in the hopes of a cure. In addition to hope, I found a supportive community, a sense of purpose, and incredibly helpful information. To me, the benefits of participating far outweigh the costs, and I can now look myself in the mirror and know that I'm doing everything I can to slow this disease, potentially for me, but more importantly for my children and for the world." And someone else taking part in the Diane TU trial commented, "It's made a real difference to me being involved in the trial. For many years I felt useless and unable to have any effect on a disease that's been rife in my family. Being part of Diane TU and having such wonderful support has helped me to grow stronger. It's enabled me to deal with many skeletons in my closet and leave them behind. The work you do goes much further and deeper than a cure." So I think we're not just talking about drugs today, we're talking about wider implications. And of course that raises many, many questions, some of which we'll raise here. I hope we'll raise more questions
and I suspect more questions than we'll be able to answer. But we're talking broadly about how society is going to have to adapt when we are faced with a condition that's so rife, suddenly becoming modifiable. We're thinking about how people can fairly make individual judgments about safety and side effects, given stigmatized views of dementia. We're thinking, what are the practical, ethical and financial implications of receiving or not receiving such treatments? We've heard, of course, that that some of these treatments will be much more easily deployed in large university hospitals, but even if you live close to one of those, what are the financial and time implications of attending for regular treatments, and will that create a sort of two-tier culture of the haves and have-nots? How will the health and third sector services need to change as we move dementia from a sort of post-diagnostic discharge phase to this more cancer-like pattern of monthly hospital visits for infusions? How will we exploit the associated opportunities for increased education and support and social participation as well? What will it mean when people are maintained for longer in these mild to moderate stages, the time when, at the moment, families often say that they need the most support? And how can we adopt a more optimistic and caring collective view? What will changing the notion of dementia being something untreatable to something that's malleable or modifiable mean for our general perceptions, not just about dementia, but about our later lives? To answer, or perhaps address or expand this list of questions, it's great to have a fantastic panel joining me. I'd love to welcome Rachel and Stephen and John Schott back to the stage, but also to introduce Kate Lee, who's Chief Executive Officer of Alzheimer's Society, and Gill Livingston, Professor of Psychiatry here at UCL and Chair of the Lancet Standing Dementia Commission and Committee. So thank you all for being here. I'm sure there will be lots of questions from the audience already. But Kate, I wondered if we might turn to you first, if that's okay, to offer perhaps a personal view or a society view on the implications of these changes and advances in disease-modifying treatment for the sort of broader dementia picture, particularly for how we offer care and support to people living with dementia.
Yeah, so I think first and foremost, it's very positive, and I was just touched by the comment made in that final session about, we all wish we were sat in this room in 20 years' time looking back on the job being done, and won't that be lovely? But from here to there is a stretch. I think that for me, Alzheimer's Society, we're in a unique position because the size and scale of Alzheimer's Society means that we touch every part of the dementia system. So right from that basic biomed research, right through to end-of-life support for families. We're working directly with about 120,000 people with a diagnosis each year. Anywhere between that kind of dementia, pre-dementia diagnosis, encouraging people to get diagnosis, right through to end stage. And we know the challenges that they face, and our strategy, Help and Hope, looks at 6 big transition points. Getting a diagnosis in the first place, coming to terms with that diagnosis and what it's going to mean. The first time people need care from outside of the home, that is a significant step for many families. Hospitalisation, the kind of implications of hospitalisation, needing an alternative home, so the point at which people need to go into residential care or to look at an alternative space, and end-of-life care. And all 6 of those big transition points are still right now, today, leading to families ending up in crisis. What we need to do is ensure that the way that we introduce these new treatments do not add a 7th crisis point to this system, because this system is creaking. I was very taken by the comment that I think a colleague up here made about what about the people that don't qualify. If I just tell you, on the announcement of Lecanemab, our support line received 1,800 calls in 1 week, 1,800 additional calls in 1 week, asking from members of the public, when will I be able to have it? Will I be entitled to it? How fast will it work? This is where my dad's up to. We're getting calls every single day, people saying, can we fly to the States for treatment? It's one of the most
common calls we take about disease modifying treatments at the moment. People saying, should we pay? We'll pay privately, can we access it? So people are desperate for hope, aligned to your quotes. And this is complex and it's nuanced. Who is going to qualify is nuanced. There's going to be a huge amount of disappointment out there for people that aren't. There's going to be a lot of guilt out there from people who didn't come forward in week 1 about their symptoms, maybe because, you know, their cultural background means that they have had family members asking them to not be public about their symptoms. And then huge amounts of guilt in the process. You know, all that exists already, and I think we've got concerns about who will manage that process. And Alzheimer's Society, you know, that's a big topic of debate for us at the moment. Is not necessarily the work that we will do in support of all of you in campaigning and lobbying government to get the system ready for these treatments, but actually really thinking about how do we co-design and co-produce workable solutions with families, fantastic people, you know, that can bring this system on in a way that means that this can be a supportive and positive process for many. But for those it isn't, how are they supported? And we're looking at kind of all sorts of different models. I'm really interested in the blood cancer watch and wait system, and I've worked in cancer for 10 years before coming into dementia, and I'm quite interested in understanding how people are psychologically supported by Blood Cancer UK, for example, during being on that watch and wait phase. Because the other thing is, as people are going through treatments, people will still be anxious about whether this is going to work. So I think for us, really thinking around some of those broader implications. The other thing that I'm going to hold my hand up and say I am personally terrified about is the UK government telling us that they can't afford social care now and are extending the period that people live with mild to moderate dementia will put an unrealistic burden on social care in the UK that we cannot afford, and that it will not be said outright, but it will be an unspoken or hidden barrier to the government approving some of these changes. And so for Alzheimer's Society, we're already talking about calling that out now. Actually, where do we stand in being stronger and more vocal about saying, we know this is what you're going to say. You wouldn't say that for any other disease. I worked in childhood cancer for 5 years. We would not tell parents that we will not treat their children because they will have long-term disabilities and be heavy users of the NHS for the next 70 years if we do. And we wouldn't have said it there, and we will not say it in dementia. So for me, I think it is an incredibly positive time. We can't get into a pit of despair about thinking it's just too hard to do. But actually, I think I am really conscious of not driving further inequalities in the system by the way that we design this. We've got a unique opportunity, knowing what we do, to kind of think about some of these solutions, co-designed, co-produced with people with dementia now. And I'm really thinking about designing an equal service. And so, yeah, huge amounts to think around, but challenging times. Alzheimer's Society really putting the full weight of our organisation to thinking around some of those bigger implications, using the fantastic network that we've got, our research network, our Regions People with Dementia, to our Dementia Voice programme, to really be that voice of patient experience and really drawing in that now and our understanding now about what that means.
So that's really helpful, and thank you for that sort of passionate view on what this means for the whole field. And it's clear from what you said from you and from the telephone calls that you've been getting and that Kath talked about every time there's any article about this, that the support needs have already begun for this long before the approval of these medications. So thank you. Great to have you. Gill, I wonder if I could turn to you next and perhaps to offer the psychiatrist's view of this field, obviously particularly...
Psychiatrist to see what psychiatrists I was very struck by what Rachel was saying about how lucky she was to fit into this very narrow band. Very few people will qualify. Most people with dementia are old. It's not just a thought, it's a truth. It's not just that most people are older, and 80% of people have two significant comorbidities when they are diagnosed. So most people will not qualify for this drug. And the huge inequities in terms of ethnicity, but there's huge inequities in terms of illness, because these are drugs that are being given to people who are physically well. And even then, there's got quite a lot of side effects. So it's a beginning, but this drug is only a beginning. And I was very struck by what John said, because I think John's absolutely right. We need to get everyone in these pathways. In our clinic, we don't discharge people with mild cognitive impairment. We see them again. Everybody is kept on an allocated, a named worker. Seen every 6 months, but they can phone up in between, and everybody is asked if they will take part in research. And since we've begun to do this, we've had a massive reduction in people admitted. They still get neuropsychiatric symptoms, but we are tackling them earlier, looking at them earlier, supporting them more. And so, I mean, I don't think anyone's done a cost modelling, but I don't think it probably costs more. A nurse is quite, a whole nurse's salary for a year would probably less than a psychiatric bed for a month. So I think it's, I think it probably is cost effective, and it means that people are around to test next round of drugs. So I think we can all work together. It's fantastic that at last a possible disease-modifying drug has had some effect, but it's not for most people. It really isn't for most people. So, you were asking about if I got this at 60, like you did 61, yeah. If I got it at 85 and I've got heart disease and other things, probably wouldn't touch the stuff. So we need a bit more to move forward, and we need to all begin to say everyone should be taken on, everyone should be followed up, everyone should have access to a named person. We should consider people psychologically, socially and medically, physically. Keeping people in good physical health and well nourished is a huge change, makes a huge difference. And we should also, and you will know that I'll say this, we also have to work very hard at tackling risk factors from earlier on to try and prevent having. So the whole huge pathways, and it's massively changed from when Nick and I started. But let's hope that it will keep changing after we finish.
Well, thank you, Jill. That's great, and particularly helpful to hear about the sort of positive changes in support and the benefits that can have that can be implementable right now. And Rachel and Stephen, I wonder if we could just turn back to you. You mentioned when you were talking with John earlier about, I got the sense that you felt very supported being part of one of these clinical trials, and that it, like perhaps a little bit like the people I quoted earlier, had made a real difference for you. It wasn't just the drugs you were getting.
Oh, absolutely. Sorry. I mean, going to get the infusions on a 4-weekly basis, and everybody's so kind in the hospital, and it's just a very positive thing, and it really helps.
Thank you. Stephen, anything?
Well, I think we said it earlier, but I'm comparing with my colleague's experience down in Brighton, who she was diagnosed with dementia on Zoom. So and has, I don't think by last December had never actually seen somebody in person. So you contrast that with us. Whatever our result might be, we were in touch with people who were interested on a monthly basis. So you're not isolated in the way that you might otherwise be. So going back to your quotes, there's a side benefit. Whether it turns out, it seems to be turning out well for us at the moment. But even if it hadn't, we would have been integrated with a kind of caring community.
Thank you. And John, I wondered if I could turn to you just briefly before we open up for questions from the audience about the role that, or the interaction between the offer of support and the therapeutic relationship you have with many of your patients and their families, and this sort of dichotomy between the haves and the have-nots that could emerge. How is either saying, sorry, you can't have this sort of treatment, or you've had this treatment but are now, for reasons of criteria, going to need to stop, potentially going to influence the quality of the relationship you have as you're trying to support those families?
So I think, as clinicians, we are discussing lots of different options with our patients all the time. There are people who can't have donepezil, or can't have memantine, or can't have a MRI scan or can't have a spinal fluid testing, or who have come to us very early or come to us very late. I think it all comes down to communication. And I think there was a point made earlier on from Heather about the importance of training and training the next generations. And I think a lot of this is going to be building the workforce, but building people who are happy and confident to be able to have these discussions with people. In every other area of medicine, there are people with cancer who will be offered treatments and people will be told, I'm very sorry, you know, those treatments aren't appropriate. The question then is, can we put them onto the right path if it's a palliative path? Can we put them onto the path that works best for them? This shouldn't be a, you know, you either get your chemotherapy or you're discharged. No, you get your chemotherapy, which is effectively what this is. Or if you can't, know that you need the support and you need the care in the community you need. So it's part of a nuanced discussion, but it can't be a binary issue. And yes, there's disappointment. I think we're all disappointed in life when we don't get what we would like to happen, whether it's in a medical sense or others. It's a matter of how it's communicated to you and having other options and support networks in place, I think.
Great. Okay.
I just want to also just kind of follow on from that point, in that I think it's really important that we remember this is a thing. It's absolutely huge and it is incredible and it has the potential to, of course, be game-changing, but it's just one thing. There is lots of other amazing stuff going on in dementia. So if I look at the applications that came in for the Longitude Prize, global applications shortlisted down to 23 mind-blowing care and tech bids. You know, technology using artificial intelligence that could compensate for cognitive decline. I mean, it's really important that for those people that don't make this, don't feel like, well, there's nothing for you. Because actually there is amazing other stuff going on, you know, the development of treatments, you know, reappropriation of existing treatments, looking at the design of care and technology, looking at care pathways, looking at our understanding of what works for people, what genuinely gets to a point at which even if someone is living with dementia, they are just living with the same kind of chronic manageable disease that they may be if they're living with diabetes. You know, we must give people hope that those things are also not a million miles off, and just because this game is in town, we haven't taken our eye off the ball of that, and amazing researchers like yourself haven't taken their eye off the ball of ensuring that there will remain improvement in dementia for everybody at any stage.
And going back to the thrombolysis element, it can be a very small change that changes the systems. Again, we have this slight negative circle. Why do we diagnose if there's not a treatment? As soon as there is a treatment, then we need to diagnose, and then if we do that, we need to monitor people at home. We get the digital tech into the home, and then we can support people remotely. So even a smallish benefit for a small number of people can be a major spur.
Jill.
I'd like to say there already is treatment for Alzheimer's disease. It's not disease modifying, but there's treatment. There's treatment for carers. There's cognitive stimulation therapy, there's donepezil, where propensity mark cohort studies have seen years on a different, a difference of 6 mini-mental state points. That's a lot over the period of time. There's already a lot of treatment and things that make a change. And what Rachel said about research, that's what everybody says. Getting people in research makes them feel better. We all like to help. Nearly all. Reciprocity is a big thing, and we know that people in research studies do better, even if they're on placebo, even if they're whatever, they do better. So there's a lot we can do already. You know, people can make decisions about their future, can do power of attorney. It's not, this is a drug or nothing. And that's a real problem. People coming in and getting a diagnosis and nothing happening is a problem now, and it should be a problem that's stopped. And people should be offered a package of things, what we can do, what we'll be able to do, and be part of this family of all of us that are going to change things in future, because there is nobody my age who, or almost nobody, who hasn't got a relative or had a relative with dementia, because it's really common. It's our problem. All of our problems as clinicians and as people. And there's a huge amount we can do now, and there will be even more in the future.
Yeah, absolutely. Time to throw it open to the audience. Some questions I think are around already. And yeah, if those with microphones can just wave in order of priority, that'd be great. Thank you.
I've just got a question from online before I do some roving, which I think is a nice extension of some of the conversations that have been having. So, as we're implementing these therapies, how can we avoid exacerbating pre-existing inequalities within the healthcare system of access and outcome?
Who would like to speak to that one? So, how can we avoid exacerbating these inequalities?
Yeah, I think currently we will be exacerbating pre-existing inequalities. But maybe we can regard it as some sort of a test of it, because currently this will not be given to people who have other illnesses. So I think we will be exacerbating them, but we can also do, like Kat said, we can be going out there and looking for people from minoritized groups and looking at them within our caseload, because we've got them coming in and offering it to them, so we can be proactive, but we will be exacerbating it because they have people with other illnesses, and the older people, and age is a inequality, is a part of the Equality Act, will be less likely to have it.
I mean, completely agree. I mean, for me, I think our research shows that 50% of the reason for diagnosis variation is rurality or financial deprivation. So, just bearing in mind that, and again, I saw this from being in children's cancer, where children can only be treated at one of a small number of treatment centres around the UK, and families that the further the distance to travel to that, direct correlation from non-compliance with treatment regimes, from families that financially couldn't afford the journeys. So for me, I think it's really, again, as Alzheimer's Society, we're really pushing ourselves to think about if we develop one from the current 10 centres, we will be pushing hard to say it needs to be available in more than that, aligned much of your thinking. But we've got to think about where people can access this support, how can we encourage and support that, how can we encourage people to support with travel arrangements, with compliance, those that are, my dad doesn't read and write. When my mum was diagnosed with Alzheimer's, they found compliance, even with simple tasks, really, really hard, because he had not been the person in the house that did the majority of that kind of booking of appointments, etc. So just again, I think it's one, being cognizant of what inequalities we could be making worse, which I completely agree. Maybe accepting we will have to do that for a while, but with a conscious eye on that can't be forever. And then thinking about organisations like Alzheimer's Society, Dementia UK, other amazing, amazing dementia organisations, tiny local ones up to big national provision. How do we work jointly with clinicians to look at how we can reduce some of those inequalities that may come from things like financial costs, travel costs, compliance, those, how do we work in that space? And how do we influence commissioners through the integrated care boards to think about commissioning services that extend fully to take into account, for example, people that are on treatment may have other costs, and of course the work that we do, and lots of us do, in stigma and stigma reduction, so that we're getting people coming forward for those diagnoses earlier, because that will remain a huge problem within various communities.
And just to push you on that one while other people think of questions, do you think the advent of these will the number of people coming forward for diagnosis or to participate in these existing treatments go up because of a general sense of hope that these treatments convey, or are there other things which will be more powerful in terms of motivating that sense that there is something that can be done? Jill?
I think it'll be extremely powerful. I think the diagnosis rates will go up because people will want a diagnosis more. Their GPs are less likely to say to them, there's no point. I think that puts people off hugely. And so I think it will massively increase the diagnosis rate. Many years ago, when we had lithium for bipolar, lots more people came forward and got the diagnosis of bipolar. I mean, in a way, diagnosis fits treatment rather than the other way around.
Yeah. John? I think in terms of what Kate was saying, Jill were both saying about the other huge advances, is that I think this comes at a perfect time
in terms of digital tech and blood-based biomarkers. And I think within a few years' time, we're going to see diagnosis rates much higher as a result of those. And I think we're also going to move from generic diagnoses to more specific diagnoses, which will then also feed into the research and the prognostication piece. So I think this may be a bit of a virtuous circle in terms of actually ascertaining and diagnosing people, which will have lots of positive knock-on effects as well.
And I think for me, it's just also part of the kind of national dialogue around dementia that these things are changing. So one of the things that I was asked recently when I spoke at something is, you know, like dementia, is that even a real thing? Is that not just like people getting old? And why are you then trying to find a drug that's about stopping people getting old? People got to die of something. And so when you say, well, actually, the bulk of the British public still believe that this is an inevitable part of getting old and is not an illness. And so actually, as we start to talk about drugs coming on board, people start to say, oh, that's weird. Why is there a drug? And so, because it's a disease of the brain. You know, why wouldn't we be trying to find a drug? And so I think it's changing the national dialogue around dementia that is really, really important as well. And so even just some of these things in the press, the headlines in the press, you know, just start to try and get us in a positive debate.
Thank you. Madam. Just hold a little closer.
Stand and shout. I think this microphone's working, so you can use this instead.
Hi. Oh, yeah. Amanda Hesgrove from UCL. I think, listening to what you're saying there, I mean, is there a case, I read recently that in schools they're looking to put subjects like menopause on the curriculum. Is there a case for putting dementia on there as well? I mean, it is a fact of life at the moment. It won't always be, we hope, but it is, you know, there are, I think, good calls for that.
Anyone? Can I talk about the last thing, about it? Maybe it won't always be. I think we very much hope there'll be treatments and there will be a compression of morbidity. But in the end, people will die of something, and it's very unlikely that all cancer, all strokes, all heart attacks, and all dementia will be cured. So yeah, I think that you know, what we hope is that people won't be having 10 years of complete dependence, and that people will be able to stop relatively early. I think that the idea that all dementia will go is over-optimistic.
Maybe,
especially as there's lots of different types of dementias.
Maybe just talk about old age then, because it is actually a stigma to get old in this in this life, I think.
I know, but it's a hell of a lot better than the alternative.
Well, yes.
My granddaughter told me recently that crocodiles don't die of anything, they just stop. And so maybe that's where we're trying to get to, is that we don't die of anything, we just stop one day.
On that note of crocodiles, any more questions before? I think I will hand back to Nick and John and Kath as co-hosts to close our meeting. But thank you again to our panel. Thank you.
So I would like to add my thanks to the panel, and particularly thank you, Rachel, again. I think we have ended up with, I think, a lot more questions than we started with, and that's a good thing. And I'm mindful, I'll keep this short because there are drinks which you're all invited to outside, and I don't want to be the person between you and drinks. It's always a bad place to be. But I just will, the fact that I've got the microphone, I'll take the opportunity to talk for a minute or two. I think there's some thanks that need to be said. I mean, I think I'm very thankful that we have these sort of prospects, and these are good problems to have, as several people have spoken about. There's a lot that we need to do, and I think it's worth us sparing a thought for all those patients who have gone into disease-modifying therapies that were negative trials for 20 years. Those are people who went through an awful lot, and then they found out that the therapy was negative. I think there's sometimes a very suspicious view of pharma. I think that my spare a thought for all those people who put efforts into all those therapies where they really devoted a huge chunk of their career to, and then they were negative. So it's a great place to be now that we have therapies, but we need to be, appreciate the people, came up with the first genetic discoveries, found that the understanding of the basic biology, those people who went into trials either as patients or helped to be site PIs or pharmaceutical companies, because this hasn't happened by chance. It doesn't just happen. And people have often said, well, why, cancer's made all this progress. Why hasn't dementia? Why hasn't Alzheimer's disease made that progress? Well, actually, cancer's been way ahead of us in terms of investment and research, and it didn't happen by chance with cancer. It was hard work and people going into trials, and that's patients and their families and the clinicians and the researchers and the multidisciplinary team. It all builds. It doesn't just happen overnight. But hopefully we're at a turning point. And the turning point, I think we've identified a whole lot of real difficulties. But if we can change that nihilism, that really has been pervasive throughout my career of what is the point of making a diagnosis for something you can't do very much about. And I think that has changed in the last few years. There are very few things that totally change the fabric of society, and dementia is one of those. It's now on the news. It's on the front page of The Guardian. It's in every element of our culture, from songs to theatre to film. And that increased awareness will help to drive, over time, better services, which we desperately need. I remember being struck very much when I used to go and see consultations on the ward in a district general hospital, that there was huge pressure not to miss a cancer diagnosis. And clinicians felt really bad. And we have 2-week waits. The opposite was true with dementia. People tried to avoid making a diagnosis because the shame was not missing the cancer, which is the shame with cancer. The shame was overdiagnosing something or making a mistake. And I think we are on the cusp of changing that mindset. And if disease-modifying therapies make some difference to that nihilism and that mindset, then that's a step forward, even if our first therapies are only available to very few. But it is hopefully the thin edge of the wedge, and we have to improve care, we have to improve diagnosis, MRI access, molecular diagnosis, all these other things, but they're good challenges. And can I invite you to thank all the speakers and have a drink. Thank you very much for coming on. The drinks are outside. I hope.